ma'am.
And I think thanks should be given to Indranil, no? Your
Yes, sir.
teacher?
Yes, ma'am. Yes, ma'am.
Yeah.
Yes, sir.
খুব ভালো একটা টপিক জানো, শুধু মত না, পাখিগুলোরও খুব কষ্ট হয় এই এত আলোতে।
হ্যাঁ ম্যাম।
আগেকার আলোগুলো ছিল কি নিচের দিকে করা, আর তার উপরে একটা গোল চাকতি থাকতো যাতে আলোটা পুরোটা এসে নিচে
ইয়েস
পড়ে,
ম্যাম।
ওপরে
এখন তো
না যায়।
আরবানাইজেশনের সাথে সাথে এগুলো বেড়েই
এখন
চলেছে।
এই যে ত্রিফলা দিয়েছে, আবার ওই উচ্চ বাতি স্তম্ভ দিয়ে এত আলো দিয়ে পাখি মথ প্রত্যেকের ডিস্টারবেন্স হচ্ছে। আচ্ছা
আর এটা
চিন্ময় কিছু
এফেক্টটা
কোয়েশ্চেন
পড়ছে ক্রপ
করবে?
প্রোডাকশনে।
চিন্ময়?
ঠিক আছে, ভালো বলেছে, সুন্দর বলেছে।
হ্যাঁ,
থ্যাংক ইউ স্যার।
কনগ্রাচুলেশন। আমরা ঠিক আছে, আরো
should I call the next participant?
ইয়েস ইয়েস, সৈপা।
Okay, Shoipa. হ্যাঁ, Shoipa Mukherjee.
থ্যাংক ইউ সৌম্য, থ্যাংক ইউ।
থ্যাংক ইউ স্যার, থ্যাংক ইউ স্যার।
হ্যাঁ, Shoipa Mukherjee, are you here? Hello, Shoipa.
Yes, ma'am.
Oh, okay. Shoipa Mukherjee from Vijay Krishna Girls College. Shoipa's topic is geographical patterns of species richness and endemism, climate versus topography. Bah. Shoipa, start.
Ma'am, is it visible?
No, Somo, off your
ইয়েস ম্যাম।
video and audio, please.
ওকে ম্যাম।
Okay. Yes. Shoipa,
Ma'am,
it is.
No.
Yes, it is visible, nicely visible. Shoipa, start.
ভেরি গুড।
Good evening to all of delegates present over here. Myself, Shoipa Mukherjee of semester 2-3, on behalf of Department of Zoology, Vijay Krishna Girls College, is going to present on the topic Geographical Patterns of Species Richness and Endemism, Climate versus Topography. Before going into further details, let us see what species diversity means. Species diversity is the number of species in a particular region or number of different species that are represented in a given community. It incorporates both the number of species in a community, that is species richness, and the evenness of species abundance. So let's see what are the components of species diversity. Species diversity has two components, species evenness and species richness. Species evenness is a measure of the relative abundance of the different species making up the richness of an area, whereas species richness is a measure of the different kinds of species inhabit in a certain area. In the figure given below, the boxes represent ecological communities, and each shape represents individual of different species. From this two figure, we can infer that community 1 has both high species richness and evenness, whereas community 2 has high species richness but low evenness. Next, see what is endemism. Endemism is ecological state of being unique to a defined geographic area. Indigenous organisms are not endemic to that particular location, even if they are found elsewhere. Extreme opposite of endemism is cosmopolitan distribution. Now, the subcategories of endemism. Endemism has two subcategories, paleoendemism and neoendemism. Paleoendemism are referred to the species that was formerly widespread but now restricted to a smaller area, whereas neoendemism are referred to those that has recently arisen, such as the species that has diverged and become reproductively isolated, or one that has formed following hybridization and is now classified as a separate species. Next, the causes of endemism. Endemism is caused by 5 causes. 1. Poor adaptability. Next, changing climatic condition, then geographical barriers, then change in land use cover or fragmentation, and then narrow ecological amplitude. Next, we see the drivers of species diversity. A number of biotic and abiotic factors have been proposed as drivers of geographic variation in species richness and endemism. Among abiotic factors, climate and topographic variables have been correlated with species richness and endemism. Main drivers of species diversity are climate and topography. In narrower aspect, both climatic variables and topographic variables influences species richness, whereas climatic variables influences endemic richness and topographic variable influences endemicity. Both endemic richness and endemicity comprises under endemism. Next, we see the first abiotic cause, that is topography. Topography is a study of the land surface. In particular, it lays the underlying foundation of a landscape. Let's see how topography influences species richness and endemism. Species richness is increased by topographic alteration or variation and reduced by the peninsular effect, in which diversity decreases as a function of distance from the main body of land. The species area hypothesis proposes that large areas contain more species than small areas because they can support larger population and a greater range of habitats. Larger populations are more resistant to extinctions. Greater ranges of habitats promote increased speciation rates, where we can see two types of topography. One is mainland and the other is oceanic island. Due to topographic alteration, we can see oceanic island has high rate of endemism but low rate of species richness, whereas in mainland, with increasing altitude, endemism increases and also there is high rate of species richness in the mainland. In this two map, we can see the species richness in North American vertebrates, which follows a polar equatorial gradient. And in this graph, we can see the declining species richness of birds and mammals at higher elevations in the Himalayan mountains. The next cause is climate. Now let's see how climate influences species richness and endemism. Here we can see two forms of climate, current climate and historic climate, which influences species richness and endemism respectively. Climate stability has been put forth as one of the main drivers of global species richness patterns because climate has a direct impact on the probability of extinction. The contemporary hypothesis suggests that the number of coexisting species varies according to current resource availability, while the historical climate hypothesis proposes that species differ in their ability to adapt to severe climate changes, and that area with the stable climate might favor long-time persistent and speciation, or by decreasing the opportunity for extinction. The impact of historical climate is probably greater on endemism, since endemic species have restricted distributions and in many cases persisted in small refugia where habitat and climate were stable for long periods of time. In this two graph, we can see the species richness is affected by the timing of rain. The first graph is of the plant richness and the second one is of the invertebrate richness. And in these four graphs, we can see the species richness of birds, mammals, amphibians, and reptiles in North America in relation to potential evapotranspiration. In these five graphs, we see the endemism per unit area by climatic fluctuations. Endemism per unit area affected by climatic fluctuation in mammals, birds, reptiles, amphibians, and vascular plants, which decreases with the increasing temperature change since the Algium, where each circle represents a geopolitical unit. Open circles represent area covered by glaciers or polar deserts during the last glacial period, whereas the filled circles represent the remaining areas. So now I want to conclude that combined influences of habit, heterogeneity, climate, and historical contingencies explain the contemporary patterns of flora and faunal diversity. Gradient and assemblage situation is likely to have influences on the potential for lowland species to shift their distributions in tracking changing climates. Dynamic natural dispersal processes could be durably compromised by habitat destruction linked to ongoing deforestation and expansion of plantation that ultimately affect the extinction and richness of endemic species. Nevertheless, the influence of historical climate, current climate, and topography are not mutually exclusive, and both species richness and endemism are likely to be the consequences of their interactions. There are some of the references which I have referred for preparing my presentation. Lastly, I would like to express my heartfelt gratitude to our principal ma'am, Dr. Ruma Bhattacharya, and our IQAC coordinator ma'am, Dr. Sweta Guha, for giving me this opportunity to present my topic in this platform. I'd also like to express my gratitude to our HOD, Dr. Papia Ghosh, my mentor, and all other faculties of Department of Zoology for their support. Thank you all for your kind attention. Thank you.
Thank you. Thank you, Soipa.
Thank you, ma'am.
Everyone, please remember that your time is 8 minutes. Huh? Thank you. Your presentation is very nice. Is there any question? Hello, Chinmoy, Obhinabo.
Not yet. ঠিক আছে, ভালো হয়েছে।
Okay. হ্যাঁ, ভালো হয়েছে, সত্যি ভালো হয়েছে।
ভালো
খুব
হয়েছে,
ভালো হয়েছে, টপিকও ভালো ছিল।
Congratulations.
I am calling the
Thank you,
third
sir.
presenter. হ্যাঁ?
ঠিক আছে, thank you, Soipa.
Thank you, sir.
Our third candidate, Diksha Chatterjee. Diksha, are you here? Hello, Diksha. Hello. Diksha is there. Can you tell me? Tanvir or Sushanto?
Diksha Chatterjee নেই।
I think Diksha is absent. Hello.
ম্যাডাম দীক্ষা অ্যাবসেন্ট মনে হচ্ছে দীক্ষা নেই।
আমারও মনে হচ্ছে দীক্ষা অ্যাবসেন্ট।
হ্যাঁ,
দেন
নেক্সট আর প্রসিড করে যান ম্যাডাম, তাহলে ও যদি পরে জয়েন করে তখন ওকে ডেকে নেব আমরা।
হ্যাঁ। নেক্সট ম্যান দেবপ্রিয় দাস, সিটি কলেজ। দেবপ্রিয় দাস ফ্রম সিটি কলেজ।
দিদি একটা বোধহয় চ্যাটে ওরা লিখেছে কথন বন্দ্যোপাধ্যায় ফ্রম সিটি কলেজ। হি উইল বি কথন বন্দ্যোপাধ্যায়।
দেবপ্রিয় অ্যাবসেন্ট।
দেবপ্রিয়র জায়গা বোধহয় কথন বলে কেউ বোধহয় রিপ্রেজেন্ট করবে।
ওকে ওয়েলকাম। ওকে কথন, your topic is tiger, a spectacular ecological bounty from nature.
Thank you very much. Am I audible?
Start. Yes, you are audible. Screen share karo. Soipa, you just off your video. Kathon, start.
Yes. I am sorry for the inconvenience. I am Kathan Bandhopadhyay. Right now, I'm a PhD candidate of the School of Biological Science of University of Reading, United Kingdom. And today we will present the different aspects of the tiger movement ecology, its prey selection, the human-tiger conflict, the tiger conservation aspects, and the Project Tiger and the challenges of the tiger conservation. Firstly, I would like to mention that tiger is one of the most apex predator of our nature, and tiger conservation requires significant conservation aspects, as it's the main flagship species as well as the keystone species of the Indian forest. And Ellsberg in 1989 predicts that the likely consequences of the removal of a top carnivore like Panthera tigris from the ecosystem are changes in relative abundance of the herbivore species with a guild and increase of the number of small predators. In the different studies like the study of the Chitwan National Park, where they radio tagged the tigress that covered the 7 to 10 kilometers per night and roughly at a rate of 0.7 kilometer per hour, male travel farther than the female, which also echoed the other studies, including the studies of the Ranthambore, the studies of the Bandhavgarh, which I participated during my undergrad from Presidency University. And according to the recent studies by the Wildlife Institute of India, Ranthambore National Park percentage of the prey selectivity relate only to the wild species that comprises 88 to 89% of the tiger diet. Multimodal likelihood ratio test, a test which you can done in our studio, and the SPSS and the Bakchi perform that test, and overall prey selection based on the availability of the prey group is significant as well as the individuals. The tiger diet were comprised of deer and swines from the wild boars, and that is the 75% of the wild ungulate crude biomass in the Indian national parks, and the proportion of the diet is expressed in the percentage. That's a good predictor of its frequency in the diet, as well as the relevant by its scat analysis. The next aspect, which is the one of the most challenging aspect of tiger conservation, that the human-tiger conflict. The human-tiger conflict possesses at least two major threats. The conflicts often results in the mortality on the removal of the tiger as a retaliation, or they result in the negative attitude of the local people towards the tiger conservation, which reduces the ability of the interactive or the interdisciplinary tiger conservation aspect. Due to the human-tiger conflict, or HTC, the tiger abundance have declined and the tiger habitats have declined roughly to 1 lakh to a few hundred individuals. Our general understanding of the risk perception and the risk analysis of HTC is required to develop an effective tiger conservation strategies. This is a review wrote by Dickman, who is now a scholar in the Wild Crew of Oxford University. According to the study of Inkins in 2003 and myself in 2018 in Bangladesh, Sundarban and Bandhavgarh National Park, proved that the less than 50% of the respondents cited the tiger problem out of the 24 to 26 relevant problems identified by the participatory risk mapping, or known as PRM process. Comprehensive wholesome plan is required to implement the presumptive, mitigative and the reactive measures, as well as the educate the community involvement components should be developed by the range states. And the food web and the tiger participation, as we all discussed earlier, that the tiger is the key component of the ecosystem, and the tiger is known as one of the most apex predator of the, especially the Bengal tiger. It promotes an upward bias of the energy flow and a downward bias of the prey selection, and controls the natural prey population. The following picture will tell the importance of the tiger in the dry forest and the dry decidual forest, where it resides collaterally with leopards and jaguars. Another aspects of the tiger conservation, which is one of the main aspects, a tiger initiative that was initiated by the government of India in collaboration with the worldwide WWF in 1973, with three specific objectives. The number one is investigating the factors that is reducing the tiger population and how the ecosystem can be repaired, and the maintenance of the sustainable tiger population. After the Project Tigers, 50 tiger reserves were run by the Project Tigers, and currently India has a percentage of 80% of the wild tigers population. Most conservative numbers of the tiger in 2018 is around 2,967, and we have to remember, when you are calculating the tigers number, we don't calculate the juvenile tigers number or the baby tigers, that because they do not appear in the camera traps or the pug mark surveys. That is the most conservative figures that we have according to the study of the WII, Wildlife Institute of India, and the Ministry of Environment, Forest and Climate Change. Tiger conservation, an international and the national wide effort to conserve tigers populations, offer an excellent example in issue in practice, found in the world, with some of the highest density and the poorest human population. And that is the main challenge of the tiger conservation, that the tiger resides collaterally in a certain demographic regions which is occupied by the poorest of human populations. Integrated conservation plans have to be promoted, as well as their livelihoods of the local people should be promoted to a sustainable tiger management practices. Comprehensive and detailed research plan indicate three variables to index to the root source of the threat of the tiger. The number one is the habitat integrity, which indices the size, the degree of the degradation, habitat fragmentation, connectivity of the habitat blocks, poaching pressure, which indices the intensify and intensification of the illegal hunting of tigers and their prey, and the potential for its control. And the third is the population status, which the indices of the tiger abundance and the recent population trends of the each tiger habitat. An extensive camera trapping and pug mark survey for identified the breeding rate of the tigress and the net survival of the juveniles. What are the challenges of tiger conservation? In a pin chart, we can mention that the 6 most challenges of the tiger conservation is: number one, the habitat fragmentation. And habitat fragmentation can lead to the inbreeding depression of the tiger, the genetic inbreeding of the tiger population do not showing a large genetic diversity in the large landscapes of the India, Malaysia, and the and other parts of the landscapes. The number two is poaching. Number three is the habitat destruction by the local people and the states and the federal agencies. Number four is attitude of the local people, which is very much important to integrate the local workers into the tiger conservation. Number five is the prey abundance. And number six, the most challenging aspect, the human tiger conflict. Tiger tourism. Tiger tourism is one of the pivotal aspects of the tiger conservation. The goal would require the investment from the both sides, inside and the outside of the tourist industry. It provides some precaution and systematic overview of a successful tiger reserve. Development of the well-equipped permanent local security forces, either police or army or armed park rangers, to locate the removal of the poacher camps and to provide security of the tour operators and tourists in the more remote parts of the forest. And boat jetties, small lodges, ecotourist camp are also vital to scrutinize the view of the overall landscape from a bird eye view on the island where tiger occurs. And the social and the political acceptance of the local people and support of the local community to develop based on the conservations and the tourist scheme. And what are the future recommendations? The future recommendations are the present status of the tiger. As we very well know, the challenges and the present status of the tiger conservation. Some future suggestions are this: plans are essential to preserve, protect, and defend the population of the indigenous tiger. A healthy tiger population supports the livelihood as well, protecting the vast landscapes where the tiger thrive, help to regulate the fresh water, reduce the impact of the climate change, and provides a source of clean air, medicinal plant, jobs, and much more. So comprehensive research plan on the tiger ecology, prey selection, prey abundance, and tiger health is mandatory. On the other hand, state and the federal government should design a framework to allow the ecotourism with the active engagement of the local communities. Through this, I would like to thank Devopriyo Das and the Devashish Kormukar, who are now studying in the Department of Zoology, City College, for this amazing topic, for choosing this amazing topic and helping a tiger fraternity of people around the globe. Thank you very much for your time.
Thank you, Kathun. Our next, Saptadipa Moitro. Kathun, your presentation is very nice. Now I am calling Saptadipa Moitro. Saptadipa, are you here? Saptadipa, Vivekananda College, Thakurpukur. Hello? Sushanto? Am I audible?
Yes, ma'am.
Yes, yes.
Saptadipa is there? Saptadipa absent?
নেই তো দেখছি।
মনে হচ্ছে সপ্তদ্বীপা নেই।
Okay. Next, Oindrila Roy. Oindrila?
Yes, ma'am, I'm present.
Oindrila?
Yes, ma'am.
Okay, Oindrila Roy of Raja Parimon College. Oindrila's topic is effect of increased salinity on growth of Sundari. Heritaria forms in Sundarban Delta, West Bengal. Very good. Oindrila, start.
Yes, ma'am.
Screen share karo, Oindrila, screen share karo. Yes, nicely visible. Start.
Okay, ma'am. Respected chairperson, esteemed professors, and learned audience present, I am Oindrila Roy, third-year outgoing student of Zoology Honours from Raja Parimon College under University of Calcutta. I am to discuss about the negative impact of increased salinity on the growth of Heritiera forms, locally known as Sundari. It is the indicator species of Sundarban mangroves situated at the river basin of Ganga, Brahmaputra and Meghna, consisting of southeast of West Bengal in India and southern part of Bangladesh. In a concise form about Sundarban mangrove is described here. The next one tells us about the systematic position, features, characteristics, conservation status, utility, and other information about Heritiera forms of Indian Sundarban. The reason of increased salinity are stated and displayed here, not entering into the details. This is very important to notice that the analysis of 3 rings of stem disc of Heritiera forms are collected at breast height from 2 different sites of contrasting salinity revealed the mean radial increase is significantly higher at low salinity than that of higher salinity. Specifically to study the effect of increased salinity on the growth of Heritiera forms, 2 remarkable works were made, one based on above ground body mass of Heritiera forms from western zone that is nearing Ganga estuary and from central zone that is Matla, Thakuran and Vidya river basin of Indian Sundarban by K. Banerjee, R.C. Gatti and A. Mitra. This work certainly established that in western zone, above ground body mass steadily falls down with increase in salinity gradient, while the total above ground body mass of central zone is very low in comparison to western zone, as the salinity gradient of this zone is much higher than the other. When the second works by A. Mitra and K. Banerjee deals with the determination of optimum salinity condition by culturing seedlings at 5 different salinity levels, those are 2, 5, 15 and 20 PSU. The effect of salinity on the pigment chlorophyll A, B, and total chlorophyll and total carotenoids are recorded over a span of 7 days, 14 days, 21 days, and 30 days. The interrelationship between salinity and selected pigments of hemipteraforms exhibited negative correlation. The results establish the optimum salinity condition for hemipteraforms is 5 to 15 pSc. These two referral experiments will help to read in the effect of salinity increase on the growth of hemipteraforms described here. Of several regions of hemipteraforms being endangered, the most common disease affecting them is top dyeing. Disease of top dyeing is not singular, but one of them is the slow increase of salinity over a long period of time. Increased salinity compensates hemipteraforms to absorb high salinity water against osmotic pressure. Even after the maximum exclusion of excess salt by species' own mechanism, high cations Na plus or H plus are balanced by high non-ionic solutes in the cytoplasm. Failure of this balancing system beyond the salt tolerance limit affects the crown of hemipteraforms, resulting to top dyeing disease of the species. Other information related to top dyeing are shown in this slide. Finally, hemipteraforms consisting the major part of Sundarban mangrove are natural ecological barrier and reduces long-term deterioration of intertidal zone, but the species itself facing threat of extinction over decades from unplanned logging at random, over deposition of sediment, in want of sufficient freshwater supply, and mostly due to increase of salinity gradient beyond its tolerance limit. So, to save hemipteraforms from major extinction, planning may be executed for preparation of favorable soil strata for seedlings of Sundari and importantly taking measures to increase the supply of freshwater by various means. This is a review presentation based on published data. Related references are listed here. We thank to the organizer for offering me the scope of this presentation as a learner.
Thank you, Indrita.
Thank you.
Moitridi.
Thank you. Thank you, Indira. Your
Thank you,
presentation is nice.
Presentation is very good.
Chinmoy?
Yes, it's good. Very good, very good.
Very good. And I'm calling the next one.
Subhro Kamal Ghosh.
Subhro Kamal Ghosh, Shoshuna College. Subhro
Yes,
Kamal, are you here?
Yes, ma'am.
Indira, thank you.
Yes, very good. Your topic is review on conservation of blue whales. Okay?
Yes, ma'am.
Start your presentation. Thank you, Indira. Thank you. No. Give 1 minute.
Sir, ma'am.
Okay. Carry on.
Okay. Good evening, sir and ma'am. I am Shubhro Kamal Ghosh from Department of Zoology, Shoshuna College. My topic is review on conservation of blue whales. First of all, the scientific name of blue whale is Balænoptera musculus. Introduction. Blue whales are mammalian balænopteran organisms and are the largest animals ever to live on our planet. It is mainly found in North Pacific and North Atlantic Ocean. It maintains a balanced marine ecosystem. They feed almost exclusively on krill, staining huge volumes of ocean water through their baleen plates, which hang from the roof of the mouth and walk like a sieve. Today, blue whales are listed as endangered under the Endangered Species Act since 1986, when it was first introduced in the list. Habitat of whales. Blue whales are found in all over the southern hemisphere.
They have been spotted off the coasts of South America, near ice shelves in Antarctica, and of the coast of Madagascar. Blue whales are also found in the warm waters of the Indian Ocean near Australia. Also in Alaska, New England, Pacific Island, North Atlantic Ocean, it has been found. আমি এখানে একটা পৃথিবীর ক্ষুদ্র সংস্করণ অর্থাৎ গ্লোবের মধ্যে ডিপ ব্লু কালার দিয়ে দেখিয়েছি কোথায় কোথায় ব্লু হোয়েল পাওয়া যায় বা যেতে। Causes of declination in numbers. The first point is vessel strikes. Vessel strikes can injure or kill blue whales. It has killed blue whales throughout their range, but the risk is much higher in some coastal areas with heavy vessel traffic. My next point is entanglement with fishing gear. Blue whales can become entangled in fishing gear, either swimming off with the gear attached or becoming anchored. Blue whales can become entangled in many different gear types, including traps, spots, or gill nets. Once entangled, blue whales may drag and swim with attached gear for long distances, ultimately resulting in fatigue, compromised feeding ability, or severe injury, which may lead to reduced reproductive success and death. Number 3 is migration. Blue whales migrate for searching of food and reproduction. Sometimes, after migration, they can't adopt the new place. Habitat degradation is shown. This habitat degradation is very much responsible for their death. Number 4 is toxic contamination. Scientists have observed that oil spills can also kill the blue whales. DDT of grassland mix into the ocean, which is most responsible for the death of the blue whales. Biodiversity many things throughout the food web, whereby top predators such as killer whales end up with extremely high concentration of pollutants within their fatty tissues. These pollutants affect on their reproduction, development, endocrine and nervous system, and also increasing their susceptibility to cancer. Some other threats are the collisions of ships, hunting for preparing blue whale oil, and illegal whaling. এই জিনিসগুলো আরো বেশি রেসপন্সিবল। আমি একটা গ্রাফ দেখিয়েছি যেখানে আমি গ্রাফের একটা ঘরকে হাজার ঘরের সমান ধরেছি এবং 1875 যখন মেনলি ওয়েলিং হতো না তখন অর্থাৎ প্রি ওয়েলিং টাইমে ব্লু ওয়েলের গ্লোবাল পপুলেশন ছিল অনেক বেশি, বাট তারপর ওয়েলিং হতে হতে তার পপুলেশন আমি গ্রাফে দেখিয়েছি কতটা কমে গেছে। এবং তারপর 1967 এ ইন্টারন্যাশনাল ওয়েলিং কমিশন যখন এই ব্লু ওয়েলকে বন্ধ করে দেয়, সেটা বন্ধ করে দেওয়ার জন্য আমাদের এই ব্লু ওয়েলিং এর যে হান্টিং ব্যাপারটা সেটা আর হয় না। এবং তারপরে এখন যা গ্লোবাল পপুলেশন ব্লু ওয়েলে সেটা হচ্ছে 10,000 থেকে 25,000, যেটা মানে মাত্র 3 থেকে 11%। অর্থাৎ 1911 সালে যে পপুলেশন ছিল, যে গ্লোবাল পপুলেশন ছিল মাত্র 3 থেকে 11%। অর্থাৎ যেটা ভালো মতো বুঝিয়ে দিচ্ছে যে আমরা ব্লু ওয়েলকে সংরক্ষণ করতে পারছি না, আমরা কনজার্ভ করতে পারছি না। Remedies. Define and undertake a long-term assessment of the number of Northwest Atlantic blue whales, the structure and trends of the population, and determine their range as well as their critical habitat with Canadian water. Reducing the anthropogenic noise like seismic exploration and disturbance from anthropogenic activities like whale watching. Availability of suitable habitat, responding to dead, injured, or entangled blue whales. Reducing vessel strikes and toxic contaminating materials, and stop the ship collision. Importance of blue whales in marine ecosystem. The blue whales play a key role in maintaining a healthy ocean ecosystem. Through their defecation, blue whales fertilize the microscopic phytoplankton upon which all sea life depends. The recovery of the Northwest Atlantic blue whale population will allow these animals, which historically were much more numerous, to fulfill their ecological role as predator and prey. Scientist Polar et al. In 2007 recently showed that Canadians are concerned about protecting blue whale for their economic benefit in St. Lawrence estuary. Conclusion: the lifespan of a blue whale is 80 to 90 years approximately, but the blue whales were injured or died before the 80 to 90 years due to the threats. In 21st century, conservation of blue whale is very much needed. The blue whale is now an endangered species. Human can save the blue whale by taking above mentioned remedial measures. They can award the Society for Conservation of Blue Whales. Human can save blue whale from to be an extinct species. This is the reference where I got some information to complete my presentation, and this is the acknowledgement. I would like to thank Dr. Indranin Saha, Associate Professor of Department of Zoology of Semiar College, for his constant guidance, advice and encouragement throughout the making of the presentation. আমি এত বড় একটা সত্যি কথা বলতে জায়গায় রিপ্রেজেন্ট করব আমি ভয় পেয়ে গেছিলাম, আমি হয়তো করতে পারতাম না, বাট উনি একমাত্র ছিলেন যিনি বলেছিলেন যে পিপিটি বানিয়ে ফেল, you will succeed. And I also would like to thank Dr. Papia Ghosh, head of the Department of Vijay Krishna Girls College, and Dr. Ruma Bhattacharya, principal of Vijay Krishna Girls College, for the marvelous organization and the fabulous mentor-mentee program. এবং আজকের জাজেসরা অর্থাৎ চিন্ময় স্যার, মৈত্রী ম্যাম, আমি আপনাদের কাছে আন্তরিকভাবে অত্যন্ত কৃতজ্ঞ আমার প্রেজেন্টেশন শোনার জন্য। থ্যাংক ইউ।
Thank you, Shubhra Kamal. Presentation is very nice. And
Very
thanks
nice
to your
and
teacher also.
very
Yes,
relevant
our next,
as well. Very relevant as well as thanks to Dr. Indranin and
yes,
Subhra Kamal also.
Thank you, sir. Thank you so much.
Thank you. Last one, actually, I want to add one talk about this. Why didn't you give your slide of importance of the blue whale at the beginning of your description? That I want to
আরেকটা কি একটু কাইন্ডলি রিপিট করবেন?
Actually, before conclusion, you have kept the slide of the importance of the blue whale. The importance of the blue whale, the slides of importance of the blue whale, you have kept just at the last portion of your description. At first, just a minute. At first, you mute yourself and. Actually, I want to know that you have kept the slide, the importance of the blue whale, at the lower portion of at the ending, at the near to the ending of your description. So I think why don't you give it? It is just a query. Why didn't you put this slide at the initiation, at the beginning of your description, the importance of the blue whale in the ecology? Okay.
স্যার, এটা অ্যাকচুয়ালি আমি একটা জিনিস ভেবেছিলাম, তাই জন্য করেছি। সেটা হচ্ছে যে আমি যখন করছি তাহলে আমি ফার্স্টে বোঝাতে চেয়েছিলাম যে ব্লু হোয়েলকে আমরা কনজার্ভ করতে পারছি বা কেন ওরা এরকম ভাবে আর কনজার্ভ হচ্ছে না, এবং লাস্টে
তুমি রেমিডিয়ালটাকে আগে দিয়ে শুরু করে
মানে আমার মনে প্রশ্ন থাকে যে কেন, কি ইম্পর্টেন্স ব্লু হোয়েলের, আমি এইটাই এর জন্য পরে দেখাতে চাই।
ওকে থ্যাংক ইউ
thank you very much, and thanks to your teacher Indranil. Our next presenter, Subhrodip Chakraborty, LJD College, Falta. Subhrodip.
Hello, ma'am.
Yes.
আমার মানে পিডিএফ টা যেটা সেন্ড করেছিল সেটা আমার টপিকটা আলাদা ছিল, আমি কি আমার টপিকটা বলবো? ওটা ফাইনাল টপিক ছিল যেটাতে আমার নাম দেওয়া ছিল।
ওকে, বলো তোমার টপিক বলো।
ওকে, থ্যাংক ইউ।
Your topic is not it is
Hi everyone, I am
ম্যাডাম ওর টপিকটা একটু ভুল আছে, অ্যাবস্ট্রাক্ট।
হ্যাঁ ঠিক আছে, you just say your topic, beta start.
Okay ma'am, thank you.
হ্যাঁ
Is the slide visible now?
Yes,
Okay.
it is visible
Okay.
nicely.
Hello everyone, I am Subhrodip Chakraborty from LJD College, Falta. As bioindicators for ecosystem degradation, a review. Alongside me is Sayan Nayak from Soshna College as a co-author to this paper. So let's begin. What is bioindicators? A biological indicator is any species or a group of species whose function, population can determine the
Subhrodip,
done in White River.
You have got some
Yes,
connectivity problem, I think. Please start again from the beginning.
Okay, sir. Am I audible now, sir?
Yes. And slowly, just speak slowly.
Okay, sir. Thank you. Hello, everyone.
Please
My topic is amphibians as bioindicators for ecosystem degradation. And alongside me is Sayan Nayak from Shoshana College, Geology Department, as the co-author to this paper. So, what is bioindicators? Well, bioindicator is any species or a group of species whose function, population, or status can reveal the health of the ecosystem. Next, where was the research being conducted and why? The research was being conducted in boreal forests of Bulgaria. Another study site was Abhijatashala Lake and Avash National Park of Ethiopia. In Bulgaria, it was done in White River and in Pukashwa National Park. And as I said above, it was in Ethiopia, it was done in Abhijatashala and Avash National Park. The reason was both these places were anthropogenically disturbed. Next, sampling or sample collection. In Bulgaria, the sampling was done by two methods. One was the covered board method, in which they used 30-inch long and 8-inch wide two-layer wooden boxes. And in Bulgaria and in Ethiopia, in pitfall trap method, they took 1 liter clear plastic cup and which was filled with 50-50 ratio of propylene glycerol and water. It was to trap invertebrates, and they did it for 2 weeks. And they analyzed the data from this covered board and the pitfall trap. Next, in Ethiopia, total number of 178 and 324 annual individuals were collected by hand from Abhijatashala Lake and Hot Spring. It was done by constrained visual encounter surveys. All potential habitats were located and studied and surveyed for approximately 2 hours. Then the collection was immersed in MS-22 solution and transferred to 10% formalin for permanent preservation in the laboratory. Next, habitat analysis. In boreal forest, each forested and regenerated plots, the structure of dominant floristics composition were analyzed in 5 stations, and each station they studied the understory as well as the overstory composition in by using Northwest ecosite type classification. At each salamander covered board in 5-meter radius, they studied the natural cover material, and for pitfall trap, they studied the natural cover material in 5. No such vegetation study were done. Only pH, water pH and water temperature has been measured. They found that the pH was 9.46 in case of Abhijatashala, temperature was 28.98 degrees Celsius, and in Hot Spring they found that the pH was 8.08 and temperature was 24.4 degrees Celsius. Next, species density calculation. In Bulgaria, the species density calculation, species-wise counting was done. In Abhijata and on Hot Spring, the species-wise density calculation was done, and they also calculated the environmental pH and the temperature, environmental factors, pH and temperature. Result. In Bulgaria, they found basically 4 salamander species. One was Plitheron cinereus, one was Ambystoma latens, then Notophthalmus viridescens, and
What happens? Shubhradeep, you are not audible.
I think there is some network issue.
Yes, I think so. Then What we have to do?
Hello.
1 second, ma'am. I can call her. Call him.
Hello.
Okay, okay.
Am I audible now, ma'am?
Yes. Beta, continue.
Ma'am, will I have to start from the first or from the air only?
From the density start.
From density, okay, sir. Okay, so species density calculation was not done in case of Bulgaria study. Since they did the cover board and the pitfall trap method, only species-wise calculated individuals were done. In Abhijata and in Hot Spring, the density calculation was done, where they also calculated the environmental factors, the mean pH and the temperature. Where result in Bulgaria, mostly 4 salamander species was done. Mostly 4 salamander species was found. One was the Pritron cinereus, then Ambystoma laterale, then Nothophthalmus viridescens, and then Ambystoma maculatus. Of which they found that Pritron cinereus was the most dominant species, and they found 2 moths of this Pritron cinereus. One was the redback moth, and the other was leadback moth. Of the leadback moths were found mostly in degenerated forests, and the redback moth was found in matured and well forests. This was a table which they presented, in which they showed the species population in matured forest as well as their ages. I'm not going into details. In Ethiopia, a total of 80 man-hours effort was done. Of which 3 species, they found 178 individuals of 3 species in Abhijata and 324 individuals of 5 species in Hot Spring. The Xerophilis xeros was the most common species in Abhijata, whereas in Abhijata, Topopterna cryptotis. And they found 2 individuals from both from this Abhijata as well as from the Hot Spring of Avash National Park to be malformed in their hind limb. Sorry, in their front limb. One was from Pacaidina ankite and the other was Xerophilis regularis. Am I audible?
Yes. Carry on.
Okay, sir.
Yes,
Okay.
carry on.
So this was okay. So this was a table which they presented where it was the number of specimens in Abhijata as well as the number of specimens in Hot Spring were less. In Abhijata it was less, whereas in Hot Spring it was more. Discussion. Amphibians they breathe through the skin. We all know that. So they are very much prone to this pollution and ecosystem degradation. The study site of White River and Pukashma National Park, the White River was going through some river development projects. So the area was little disturbed. Whereas compared to Pukashma National Park, it was a well-maintained forest. So the population of salamanders in the Pukashma were of more as compared to the populations in White River was less. And also they found the leadback moths of Pritron cinereus to be more in the White River, whereas the redback moths were more in Pukashma National Park. So this in the study, Abhijata Sala Lake, they found more populations of anurans because they found less populations of anurans because the study site is disturbed. It was the survey was done in 2000 that the water level has decreased to 5 meters over 3 decades, and so it is more susceptible to climate change. Whereas as compared to the hot springs of this hot springs of Avash National Park, which was less disturbed, so they found more anurans population there. Scope for future, I think the research must also be done on Sicilians. We always forget this order Apoda, and so I guess it will give a 360 view of amphibians as a high indicator for ecosystem degrading. Thank you. These are the references which I took. Thank you for bearing all these speeches of peace of mind and all these problems which took place. And I want to thank Biodiversity Girls College for giving me the opportunity to speak. Thank you.
Thanks. Thanks a lot. Nice presentation. And thanks to your teacher also. Now, Chinmay, will you say anything?
No, it's okay. Good. ও একটুখানি তাড়াহুড়ো করে ফেলছিল, এটাই আস্তে আস্তে করবে, এত টেনশন নেওয়ার কিচ্ছু নেই।
হ্যাঁ, আসলে প্রথম তো বাচ্চা ছেলে সব। হ্যাঁ, নেক্সট হচ্ছে শতদ্রু দাস, সরসুনা কলেজ। Shotadru, are you here?
Yes, ma'am.
Yes. Your topic is mangrove as an unique ecosystem.
Yes,
Traits,
ma'am.
importance, and their protection. Very good.
Yes, ma'am.
Carry on. Start.
Ma'am, is the presentation is showing?
Yes, yes. I mean, not
Yes,
yet.
yes, it is visible. Yes.
Okay.
Start better.
Good evening, everyone. I am Shotadru Das, Part 3 undergraduate student of the Department of Zoology from Shorsuna College. And the topic for my presentation is mangrove as an unique ecosystem, threats, importance, and their protection. First, I have given a file picture of a mangrove forest. Introduction. The term mangrove refers to a tidally influenced wetland ecosystem with the intertidal zone of tropical and subtropical latitudes. In other words, a mangrove is a shrub or a small tree that grows in brackish water or saline water. The distribution of mangrove ranges between 25 degrees north to 25 degrees south. Mangroves are found in depositional coastal plains where fine sediments, often with high organic content, collect in areas protected from high energy wave actions. Threats to mangrove ecosystem. First, clearing. Mangrove forests have often been seen unproductive and smelly, and so cleared to make room for our agricultural lands, human settlements, and infrastructure such as harbors and industrial areas. Second, over harvesting. Mangrove trees are used for firewood, construction wood, wood chip, and pulp production, charcoal production, and as animal fodder. In some parts of the world, it is no longer sustainable, threatening the future of these forests. River changes, dams, and irrigation reduce the amount of water reaching mangrove forests, changing the salinity level of water in the forests. If the salinity becomes too high, the mangroves cannot survive. Freshwater diversions can lead to mangrove drying out. In addition, increased erosion due to the land deforestation can massively increase the amount of sediments in the rivers. Overfishing. The global overfishing crisis facing the world's oceans has effects for beyond the directly overfished populations. The ecological balance of the food chains and mangrove fish communities can also be altered. Pollution, fertilizers, pesticides, and other toxic man-made chemicals carried by the river system from sources upstream can kill animals living in the mangrove forest, while oil pollution can smother the mangrove forest and suffocate the trees. Climatic changes. Mangrove forests require stable sea level for long-term survival. They are therefore exactly sensitive to the current rising sea levels caused by the global warming and the climate change. Importance of the mangrove ecosystem. Fisheries. Mangrove forests are home to a large variety of fish, crab, shrimp, and mollusk species. These fisheries form an essential source of food for the thousands of coastal communities. Timber and plant products. Mangrove wood is resistant to rot and insects, making it extremely valuable. Many coastal and indigenous communities rely on this wood for the construction materials as well as fuel. These communities also collect medicinal plants from the mangrove ecosystem and use mangrove leaves as the animal fodder. Coastal protection. The dense root system of the mangrove forest traps sediments flowing down rivers and off the land. This helps stabilizes the coastal lines and prevent erosion from the waves and storms. In areas where the mangroves have been cleared, coastal damage from the hurricanes and typhoons is much more severe. Wildlife habitat. The mangrove system supports a range of wildlife species including crocodiles, birds, tigers, deers, monkeys, and honeybees. Many animals find shelter either in the roots or the branches of the mangroves. The mangroves serve as rookeries or nesting areas for the coastal birds as well. Improving the coastal water quality. The mangroves maintain coastal water quality by abiotic and biotic retention, removal and recycling of the nutrients and pollutants and particulate matter from the land-based sources, filtering these materials from the water before they reach the seagrass habitats. Mangrove root system slow water flow, facilitating the deposition of the sediments. The mangroves supply nutrients to the seagrass community, sustaining these habitats' primary production and the general health. The need to conserve the mangrove ecosystem. When we destroy the mangroves, not only do we destroy the ecosystem that function naturally, we also lose the free services the mangroves provide us with and will have to pay the price to restore them. If the mangroves are destroyed, there will be degradation of the ecosystem and species endangerment as well as the loss of the wetlands. Fish and wildlife population will also decline. Floods and other water problems will also arise. Thus, there is a need to conserve and restore this ecosystem to its former glory. The conservation procedures, they are afforestation, legislation, monitoring and conducting of the surveys, protection, recommendations, soil conservation, and the status of the conducted studies and surveys. Conclusion. The mangrove forests are one of the world's most threatened tropical ecosystems. Threats to mangrove forests and their habitat includes clearing, overharvesting, changes in the river system, excess of fishing, changes in the climate, etc. Mangrove forests are extremely productive ecosystems that provide numerous good and serves both to the marine environment and people. Mangrove forests are home to a large variety of fish, crab, shrimp, and mollusk species. The mangrove system supports a range of wildlife species, including crocodiles, birds, deers, and others. Protection of the mangrove forest helps to preserve biodiversity as well as helps in protecting a vital resource for the local communities. Here are the references which I have taken for doing my presentation. Thank you.
Thank you, Shatadru, for your nice
you,
presentation, and thanks to your teacher, Indranil, also. Okay.
Shatadru,
Chinmay?
one thing, from the next time, you try to recapitalize or memorize the themes. Actually, the reading from the slides is not a good habit. Actually, you have to convince the audience, Also, from the next time, you have to practice this.
Okay, sure, sir. Thank you. For
আসলে জানিস তো, হ্যালো শতদ্রু, পড়ানো বক্তৃতা দেওয়া এগুলো কিন্তু সব রিহার্সালের প্রয়োজন হয়, যেটা স্যার বললেন। বারবার প্র্যাকটিস করলে তবে তোর মাথার মধ্যে ওটা গাঁথবে, তবে বোঝা যাবে তুই ওটাকে নিজে কনজিউম করেছিস। নিজে কনজিউম করতে না পারলে সেটা ঠিকঠাক সবাইকে দেওয়া যায় না। কেমন?
ভালো হয়েছে, কংগ্রাচুলেশন। আরো ভালো হবে।
হ্যাঁ, আরো ভালো হবে। ঠিক আছে। নেক্সট, শতরূপা সাহা। আর ইউ হিয়ার? শতরূপা অ্যাবসেন্ট? সুশান্ত?
শতরূপা তো নেই দেখছি।
হ্যালো?
হ্যাঁ ম্যাডাম, শতরূপাকে দেখতে পাচ্ছি না ম্যাডাম।
আমার মনে হয় শতরূপা অ্যাবসেন্ট। Next, Sriparna Chattopadhyay of Ashutosh College. Sriparna, are you here?
Yes, ma'am. Ma'am, I'm audible.
Okay. Welcome, beta. Yes, your topic is impacts of ocean acidification and global climatic changes on coral ecosystem and approaches to abate it. Very good. A review. Sriparna, start.
Yes, ma'am.
Start. Screen share karo. Yes.
Ma'am, is this visible?
Coming.
Yes, it is visible.
Okay. Shall I start?
Yes, obviously.
Yes, start.
A very good evening to everybody present here. I am Shriparna Chattopadhyay, and UG student of Ashutosh College, Zoology Department, and I am here to present my topic, that is impacts of ocean acidification on coral ecosystem, coral bleaching, and approaches to abate it. Let us dive into the colorful world of corals. Biodiversity is the way, so don't let nature go astray. With this quotation, I will go directly into my topic due to the time constraint. Now, let's move on towards one of the most important biodiversity hotspots on Earth, that is coral reef. So, what are coral reefs? Coral reefs are large underwater structures composed of skeletons of colonial marine invertebrates called coral. These are some of the most diverse ecosystems in the world. Each individual coral is referred to as a polyp. Coral polyps live on calcium exoskeletons of their ancestors, adding their own exoskeleton to the existing coral structure. Thousands of species of corals have been discovered. Some live in warm, shallow tropical seas and others in cold, dark depths of ocean. The largest of the coral reef system is a Great Barrier Reef in Australia, which is more than 1,500 miles long. Now, let's know about some of the types of the coral reef. Let us see the pictures. Okay. So here, scientists have basically divided coral reefs into 4 classes. First is a fringing reef, which grows near the coastline around islands and continents. They are the most common type we see. Next, the barrier reef grows parallel to the coastline but are separated by deeper, wider lagoons. The Great Barrier Reef in Australia is the largest and famous of all the barrier reef in the world. Next comes the atoll. Here we can see these are rings of coral that create protected lagoons and are usually located in the middle of the sea. Next, finally the patch reefs. These are small isolated reefs that grow up from the open bottom of the island platform or continental shelf. Now the point, why scientists, ecologists are so concerned about coral reefs. Why are coral reefs so important? First of all, it gives a coastal protection. They absorb waves energy and contribute to environmental protection to the reduction of coastal erosion. They reduce the damage in case of storms, hurricanes, and in some way the energy of tsunamis. Second, as inhabited, it is one of the most important biodiversity hotspots on Earth. They are the basis for the formation of other ecosystems too. Third, as a food resource, reef animals are an important source of protein, contributing about a quarter of fish catch average in these countries. Fourth, an economic importance, millions of people around the world depend on coral reefs for food, protection, and employment. Fifth, as tourism wealth. And obviously, sixth one, the corals promises a medical future. They are used in prevention from breast cancer, ovarian cancer. They have anti-tumor properties too. So from this glimpse, we can understand how significant coral reefs are to our biodiversity and humanity too. But now, coral reefs are dying around the world. Major threats to coral reefs are there. And I will not go to the details as we all know about it. Climate change, ocean acidification. Here we can see the picture which shows how tragically the beautifully colored corals have turned into a dying whitish skeleton. Some other factors, pollution, sedimentation, coral mining, careless tourism, and obviously overfishing. All these factors serve as great threat to corals. Now I focus on the cause my review contains, that is ocean acidification and coral bleaching. So what is ocean acidification? Ocean acidification, or OA, refers to a reduction in the pH of ocean over an extended period of time, caused primarily by the uptake of the carbon dioxide from the atmosphere. When CO2 is absorbed by the seawater, a series of chemical reactions occur, resulting in an increased concentration of hydrogen ions. This increase causes the seawater to become more acidic and causes carbonate ions to be relatively less abundant. And we know how carbonate ions are an important building block for the structures such as seashells and coral skeletons. So we can know what the thing can be. Next, here I will show here healthy versus unhealthy coral reef. We can see how tragically this is transformed and an effect of ocean acidification on corals. I will move to what is coral bleaching. The stunning colors in corals come from a marine algae called zooxanthellae, which live inside their tissues. When corals get stressed from things such as heat, light pollution, they react by expelling this algae, leaving a ghostly transparent skeleton behind, and this is coral bleaching. To maintain reefs net growth or net natural condition, curbing impact of OA is therefore very necessary. So a variety of mitigation and strategies have been taken. Some of them are conventional, like spatial planning, reducing fishing pressure on herbivores, reef restoration, to some unconventional novel techniques such as phytoremediation, chemical remediation, assisted evolution. Most of the adaptation-focused interventions use ecological mechanisms to enhance ecosystem resilience, that is ecosystem restoration. And some of the passive techniques are there, which refers to those which remove sources of stress to prevent natural recovery. Now we will go to explore a new strategy, that is marine probiotics, increasing coral resistance to bleaching through microbiome manipulation. So the first question comes is, what are probiotics? Probiotics are live microorganisms promoted with claims that they provide health benefits when consumed, generally by improving or restoring the gut flora. Of course, when most of us hear about probiotics, we think about a supplement or gut health. Indeed, the same principles can be applied to coral reefs too. A new attempt has thus been taken by the researchers to extrapolate the widespread and well-established use of bacterial consortia to protect or improve health in other organisms to corals. The attempt is manipulation of the coral-associated microbiome was facilitated through addition of a consortium of native isolated from Pocinopora damicornis, known as putatively beneficial microorganisms for corals, that is PMBC, BCL, including some of the related species strains. What was done was that from a controlled aquarium experiment in two temperature regimes, that is 26 and 30 degrees centigrade, and 4 treatments were done. That is one with PMBC, second with PMBC with pathogen challenge. Pathogen was Vibrio coralli liticus. Third is VC pathogen challenge, and fourth is controlled. This revealed the ability of the PMBC consortium to partially mitigate coral bleaching. Significantly reduced coral bleaching metrics were observed in PMBC inoculated corals in contrast to controls without PMBC addition. Here I come to a picture which depicts the processes and strategies for identifying and application of beneficial microorganisms for corals for increasing coral resilience. I will not go to the details of how to isolate the microbes and further processes, but rather want to draw your kind attention towards the application path. That is there. Here we can see the BMC consortium is applied in aquarium or reefs before a bleaching effect. And in the first case, we can see that due to the application of BMC consortium, the bleaching effect was much lesser when increase in temperature was there compared to the second case where there is much more bleaching effect, as no application of BMC consortium was there in the second case. So the results indicate that the microbiome in corals can be manipulated to lessen the effect of bleaching, thus helping to alleviate temperature stress too. Finally, the conclusion, I've concluded 3 points. That is, several strategies are there to curb the impacts of ocean acidification. Some are conventional, I have mentioned, and some are unconventional, like phytoremediation, assisted evolution, and the new one, that is the marine probiotics, which increases coral resistance to bleaching through microbiome manipulation. Finally, I would like to end again with a quotation. That is, biodiversity starts in the distant past and it points towards the future. So this is us who have to get it in the neck and have to conserve them. Here are some references I took which helped me to complete my abstract and presentation. And finally, I would like to express my wholehearted gratitude to Principal of BKGC Dr. Ruma Bhattacharya ma'am and IQAC Coordinator Dr. Sweta Kuku ma'am to provide me this opportunity to present my topic in this platform. I would like to extend my heartfelt gratitude to Dr. Papia Ghosh ma'am, HOD of BKGC, for her constant guidance, support and encouragement. Last but not the least, I'm thankful to Dr. Sriparna Datta Roy, HOD of my college, Ashutosh College, and Dr. Deep Chandra Chakraborty for guiding me. Thank you all.
Thank you, Sriparna.
Welcome, ma'am.
My earnest request to all the candidates, please maintain the time. It is only 7 minutes to 8. Please. Your presentation is nice, and thanks to your teachers also. Chinmoy, will you say anything?
No. It's a very good and nice presentation. Thank you. Congratulations.
Thank you.
Yes. Now, our next candidate,
Sujata Duari.
Sujata Duari, Botany Department, Vijay Krishna Girls College. Sujata, are you here?
Yes, ma'am.
Oh, okay. Your topic is endemic plants of India and their conservation. Okay, okay. It's a broad topic. Carry on. Beta start. Hello, Sujata.
Ma'am, is it visible?
Yes, visible. Yes, start.
Okay, ma'am. Good evening, everyone. I'm Sujata Duari, Department of Botany from Vijay Krishna Girls College, Howrah. Today, I'm going to discuss the topic about endemic plant species of India and their conservation. So let's start. First of all, we have to know what is endemic species. Endemic species are those that are found in just one geographical region. They are important because of their restriction to a particular area due to climate change and their occurrence. Characters. Number 1, they have a narrow geographical range. Number 2, with one or few population remaining. Number 3, they have small population size and genetic variability. And number 4, usually overexploited. Endemism in India. In India, there are about 17,000 species, of which 5,725 are broadly considered as endemic, that is 33.67%. India has 3 mega centers of endemism, that is Eastern Himalaya, Western Himalaya, and Western Ghat. In Himalaya, there are about 3,771 endemic species, that is 20.41%. In peninsular India, there are 2,015 endemic species, that is 18.85%. Andaman and Nicobar Island have 239 endemic species, that is 1.40%. India, however, rich in flora with fairly high degree of endemism. In India, there are about 141 flowering genera of endemic species. Those genera are highly restricted in some major regions, that is in Himalaya and peninsular India. This slide presents the graphical representation of the 3 mega centers of India, that is Himalaya, peninsular India, and Andaman and Nicobar Island. In this slide, we know about some Indian identified endemic region of the mega centers. In Agasthyamala Hills, there are about 189 endemic species. In Himalaya, there are about 183 species. In Nilgiri Silent Valley, 150 species are present. In Andaman group of islands, there are 132 species, and in Andamalai High Range, there are about 94 endemic species present. Endemism in peninsular India. In detailed studies, we found that in peninsular India, there are about 1,932 taxa, including 1,788 taxa in species level and 144 taxa in intraspecific level. And this picture presents the longitude and latitude degree of peninsular India. In India, out of 18 genera of endemic grasses, about 13 are found in peninsular India. Some of them are Vidia, Danthonidium, Indopoa, Limnopoa, Normanboria, etc. And in this slide, some endemic plant species are presented. Endemism in Western and Eastern Ghat region. The major concentration of endemic mostly found in Western Ghat region. 76 taxa are endemic to Western Ghat region. There are about 2,015 endemic taxa, among them 1,425 belongs to dicotyledons and 590 taxa belongs to monocotyledons. And mostly common families are Poaceae, Rubiaceae, Acanthaceae, Orchidaceae, and Fabaceae. Endemic genera in India. In India, there are 141 endemic genera. Acanthaceae and Poaceae have the highest number of endemic genera, 17 in each family, 114 endemic species regarded as monotype, 15 genera are widely distributed in India, namely Glossocardia family in Compositae, Octotropis family Rubiaceae, etc. These are the pictures of some endemic plant species of India. List of some families according to their percentage. Lauriaceae family have 163 taxa with 133 endemic taxa along with 80.9%. Umbelliferae have 209 taxa with 165 endemic taxa with 78.8%. Ranunculaceae have 180 taxa with 135 endemic taxa along with 75% of endemic. Roseaceae family have 250 taxa with 182 endemic taxa with 72.8% of endemic. Balsaminaceae family have 180 taxa with 123 endemic taxa with 68.3% of endemic. And lastly, Acanthaceae family have 380 taxa with 224 endemic taxa with 50.9% of endemic. The most common endemic plant species of India are Crotalaria, Juncea, Ficus religiosa, and Eleusine coracona. Factors of endemism. The factors that are responsible for the origin of endemic species is natural crossbreeding and mutable environment. Some natural barriers such as mountain, sea also lead to form endemic plants, and some abiotic factors like temperature, humidity also respond for endemism. Importance of endemic plant species. Endemic species are mainly restricted in distribution. Endemic species carry more risk of extinction than broadly distributed species. Endemic species are often endangered, so it is very important to save the species. Endemic species run a higher risk of extinction because of their geographical isolation. Conservation. Endemic species are often the most vulnerable to anthropogenic threats due to their unique evolutionary history and relatively low population size. The conservation of endemic species is a global priority, and we all are committed to preserving those. Conclusion. In India, endemic plant species are mainly concentrated in Himalaya, peninsular India, and Andaman and Nicobar Island. According to their endemism rate and floral diversity, it is very essential to conserve the endemic plant species by Forest Department. Bibliography. In this slide, there are some references that helps me to collect all of the information. And lastly, I would thankful to Dr. Pamela Shaha for helping me to do such a presentation, and also thankful to our HOD Dr. Papiya Ghosh and our principal Dr. Ruma Bhattacharya and all other judges for organizing a beautiful platform. Thanking you.
হ্যালো দিদি আছেন? হ্যালো, ওকে, সুজাতা।
হ্যালো, হ্যাঁ হ্যাঁ, সুজাতা, ভেরি নাইস। হ্যাঁ, চিন্ময় বল।
Actually, Sujata, I didn't find any conservation strategies regarding the conservation of these endemic plants. এই জায়গাটা একটু তুমি একটু এন্ডেমিক প্ল্যান্ট সম্বন্ধে, তার ডিস্ট্রিবিউশন সম্বন্ধে বলেছো, কিন্তু তার টপিকে কনজারভেশন স্ট্র্যাটেজি সম্বন্ধে আর একটু দিলে ভালো হতো। তো যাই হোক,
মানে কি করতে হবে, এই যে কি ভাবছি আমরা, কিভাবে করব, সেইটা একটুখানি দেখানোর দরকার ছিল। ঠিকই আছে, যাকে ওরা তো এখন বাচ্চা, এগুলো একটু ভাববে। হ্যাঁ, স্যার যেটা বললেন, সুজাতা স্যার যেটা বললেন, এটা একটু ভাববে, কেমন? ওকে। নেক্সট দিশানি নাগ, তাই তো? বটানি ডিপার্টমেন্ট, বিজয় কৃষ্ণ গার্লস কলেজ। দিশানি, are you here?
Yes, ma'am.
Yes. Your topic is plant diversity. of Kutch, Gujarat. Okay, carry on. Start.
Yes, ma'am.
Yes, this one it is visible. Start.
Okay, ma'am. Good evening, respected professors and everyone. I am Dishani Nag, the student of second semester Botany Honors of Vijay Krishna Girls College. So today, the topic of my discussion is plant diversity of Kutch, situated in Gujarat. So let's move into the topic. So, plant diversity. Are mostly photosynthetic organisms with plants. Plants are also marked as key segments with wide range of significance in the formation of global diversity. Plant biodiversity is very vital to ecosystem food crop and medicine production because it balances the ecosystem, protects the watersheds, moderates the climate, and provides shelter for the animals. There are four types of diversities. Those are diversity, species diversity, ecosystem diversity, and molecular diversity, resulting in diversity of ecological habitats ranging from tropical, subtropical, temperate allied to desert. There are 5 types of ecosystems. Those are desert ecosystem, forest ecosystem, grasslands ecosystem, precipitation land, which are largely barren except the seasonal vegetation cover. Covering of 2% land mass in India are classified into 3 distinct types. Those are the desert, and next one is the high altitude cold desert of Jammu and Kashmir, and the last one is the salt desert of Kutch of Gujarat. So under the salt desert, my topic is Ran of Kutch. Kutch is the district of Gujarat state in western India with its capital at Bhuj. It covers an area of 55,674 kilometers square. As we all know, it is the largest district of India. The Ran of Kutch is known for its marshy salt flats as well as the ecologically important Banni grasslands with their seasonal marshy wetlands, which form the outer belt of the Ran of Kutch. So in the geographical situation, we all can see the map. Kutch district is surrounded by the Gulf of Kutch and the Arabian Sea in the southern and the western part, while the northern and eastern parts are surrounded by the Great and Little Ran of Kutch. Next is Great Ran of Kutch. Great Ran of Kutch is one of the most remarkable and unique region in the entire world due to its wilderness value and appears like a tabletop surface, which is interspersed with the small islands locally called as Dhoi. The island supports many important species of flora and fauna. In Great Ran of Kutch, Pancham is the largest island, and in 2002, being a high rural diversity area, it was suggested that it should be declared as an ecologically sensitive area. Some rare and endangered plant species of Great Ran of Kutch is Ephedra foliata, Fida tiagi, Pavona sattakarpa, etc. Next is Little Ran of Kutch. The Little Ran of Kutch is the southward range of the Great Ran of Kutch, which is usually described as a flat, saline waste or soil-impregnated wilderness. Total 74 islands have been identified, out of which 51 having the vegetation cover. In Little Ran of Kutch, totally 253 flowering plant species have been enlisted. Little Ran of Kutch unique components of both the flora and fauna. According to the data available from Red Data Book of Indian Plants, many rare and narrow sections of plant species of Gujarat are found in the Little Ran of Kutch. For example, Urochandrum petulosa, Moringa, etc. During the survey vegetation of the Little Ran of Kutch recorded about 108 plant species distributed in the area under the study. Out of them, 16 plant species are the ecologically important plant species, Some of them are Presetatio, Delonix elata, Acacia nilotica, etc., which belong to many dominant families like Poaceae, Mimosaceae, Chenopodiaceae, etc. These plant species have several ecological important values like soil erosion, decreasing salinity, waterlogging, etc. And 49 plant species are the life-supporting plant species. Some of them are Digitaria ciliaris, Moringa olivera, Fagonia critica, etc. These plant species have several important life-supporting features like fluid, gum, honey, crops, seeds, etc. Next is the most important part of my today's speak, that is diversity of species. According to US Congress Office of Technology Assessment, there are roughly 250,000 species of land plants currently extinct. Of those, roughly 60,000 plant species are thought to possess some food value for the humans, and only about 150 plant species have been commercially cultivated. In 1974, only 7 of the 30 major cultivated crops had harvest in excess of the 100 million tons. Those are wheat, rice, corn, potato, barley, sweet potato, and cassava. Many minor crops are also in danger of the economic extinction. Some of them are relative of food wheat, for example, Cannabalia regalis, cassava, and the most important is African ahab nut. So, we can conclude that knowledge on the ecologically important and life-supporting species, which is an outcome of the experience passed on to successive generations, make the desert dwellers patiently, the rural poor to survive not only in the normal harsh desert environment, but also in the prolonged drought and famine. It is also paramount importance that advanced planning is made to preserve and propagate all the ecologically and life-supporting species to meet to experiences of the future. So here are some references from which I have collected the information of my topic. And lastly, I would like to thank our respected principal, Ma'am Dr. Ruma Bhattacharya, and respected HOD, Ma'am Dr. Apia Ghosh, for organizing such a program and give me a chance to prepare myself for this presentation. And I would also like to express my heartfelt gratitude to Dr. Pamela Shaha, who is my mentor, and all the respected professors of the Department of Botany of Vijay Krishna Girls College, Howrah, for helping me to prepare this presentation, as I came to know about lots of important and interesting facts about the topic biodiversity. Thank you all.
Thanks. Thanks, Dishani. Chinmoy?
Thank you, ma'am.
Presentation of the slide as well as the speaking capability was good.
Yes.
Thank you, sir.
Should I call the next one, Chinmoy?
Yes, it's Arindam Mukherjee.
Arindam Mukherjee, microbiology. The
Excuse me,
Joint College.
Yes, yes.
I am from his college only, Abhishek sir. Actually, he won't be able to present today, I think, because he is having network issues.
Oh, Arindam Mukherjee is not available to, not present today, no?
Okay.
Oh,
Then
Indira Das.
Oindrila Das. Vijay
Next.
Krishna Girls College, microbiology. Oindrila. Hello, Oindrila. Hello.
Oindrila, are you here?
Oindrila.
Perhaps
No, Oindrila, net problem or anything.
তাহলে অভিষেককে ডাকা হোক।
Now Abhishek. Yes, you are present here.
ওয়েন্দ্রিলা নেই
Abhishek
ম্যাডাম,
Shah. Huh?
ওয়েন্দ্রিলা নেই, বেরিয়ে গেছে সম্ভবত।
Abhishek Shah from Joint College. Abhishek's topic is genomic biodiversity of coronaviruses. Reveals natural origin of SARS-CoV-2. Okay, beta, carry on.
hello everyone, this is Abhishek Rao from PhD Jan College. I am here to present my paper on the topic entitled Genomic Biodiversity of Coronaviruses Reveals Natural Origin of SARS-CoV-2. Since 2019, the entire world has been ravaged by a single virus, which is spreading like a wildfire, infecting more than 22 million people worldwide. Modern world, for the first time, is facing a pandemic of this large scale, and hence there are expectations that the causative agent of this disease COVID-19, the virus SARS-CoV-2, may be genetically engineered. The virus has created havoc in almost all the countries, be it developed or developing countries. So let's see whether SARS-CoV-2 is natural or it's man-made. The novel coronavirus SARS-CoV-2 belongs to coronavirus family of viruses, which have RNA as their genetic material that causes respiratory tract infections in mammals and birds. The coronaviruses can be classified as alpha, beta, A, B, C, D, gamma, and delta coronaviruses. SARS-CoV-2 belongs to beta family of coronaviruses. However, only 7 coronaviruses are known to infect humans. HKU1, OC43-229E, and NL63 cause mild symptoms, whereas SARS-CoV, MERS-CoV, and SARS-CoV-2 have caused epidemic in this 21st century. The novel coronavirus SARS-CoV-2 is much more contagious than SARS or MERS. However, it's far less deadly than the other two. Receptor recognition is an important determinant of coronavirus infection and pathogenesis. With regard to RBD, that is receptor binding domain, SARS-CoV-2's RBD has much greater human ACE2 binding affinity than SARS-CoV RBD does. However, the entire RBD can be either a standing up state, which enables receptor binding, such as in SARS-CoV, or a lying down state, which helps for immune evasion, such as in SARS-CoV-2. Viruses commonly hide their RBD or other critical parts of spike protein from host adaptive immune response. However, the affinity for human ACE2 is much more, which acts as a compensation for hidden RBD. To maintain high infectivity, SARS-CoV-2 activates host protease 2. Such an efficient RBD can be achieved by natural means only. In this picture, you can see the red colored portion is the RBD, which is involved in ACE2 binding. It shows hidden RBD, which helps in immune evasion. Computational analysis shows that both the RBD are different. Now, SARS-CoV-2 has a S1, S2 cleavage site between S1 and S2 junction. With proline in it, it's known as PRAR site. It has also 3 adjacent O-linked glycans, which helps to cleave it by several proteases. Without polybasic cleavage site, infection is of low pathogenicity. Furin cleaves SARS-CoV-2 but not SARS-CoV, which is important in the activation of SARS-CoV-2. Now, an experiment was done fluorogenic peptide mimetics of S1, S2 spike cleavage site of SARS-CoV and SARS-CoV-2. These were incubated with several proteases. Using a fluorometer, velocity was mentioned. Now, we can see that matripase and PC1 and even trypsin2 cleave effectively and recognize SARS-CoV-2 S1, S2 cleavage site. These data confirm that SARS-CoV-2 entry was dependent on lysosomal cathepsins. SARS-CoV-2 has substantially expanded its activation profile with various activation from proteases. Now, not only lysosomal cathepsins, but SARS-CoV-2 is also dependent on cell surface expressed TTSPS such as TMPRSS2. Now, S1, S2 in furin cleavage, it also reflects the more exposure of cleavage site to TMPRSS2 and cathepsin. Overall, novel S1, S2 insert is likely to enhance spike protein cleavage by multiple proteases. SARS-CoV-2 arose by mutation or recombination. Now we have compared SARS-CoV-2 with several other viruses. Comparative study shows that the polybasic cleavage site and even the O-linked glycans was not seen previously in lineage B of beta coronaviruses. Although HQ1, which is in lineage A of beta coronaviruses, HQ1 has those sites. SARS-CoV shows 79% homology with SARS-CoV-2, whereas RAG-G13 coronaviruses, Rhinocephalus affinis bat, shows 96% similarity. Although its RBD differs, it suggests that its inability to bind with human ACE2. With coordinates based on SARS-CoV, there are Y442, L472, N479, D480, T487, Y491. These corresponds to the SARS-CoV-2's L45, L455, F486, Q493, S494, N501, and Y05 in SARS-CoV-2. 5 out of 6 residues differ. Now we have compared coronaviruses with other coronaviruses too, such as RMNYO2 from R. malinus, which exhibits 93.3% sequence identity in most genomic regions, such as 97.2% identical in longest gene encoding region 1AB, but in S gene it only shows 7.8 nucleotide similarity. In RBD region, RM&Y2 possess 62.45% similarity, whereas pangolin beta coronaviruses has amino acid identity of 97.4%. Two loop deletions in RBD may reduce the binding of RM&Y2 with ACE2. RM&Y2 contains an insertion at S1S2 cleavage site in spike protein. This suggests that the SARS-CoV-2 may have evolved by a mutation because it can bind with human ACE2 but not RM&Y2. Now we have compared RBM model of different coronaviruses. The red color in this shows the amino acid homology with SARS-CoV-2, and the blue color shows the differences with SARS-CoV-2. SARS-CoV-2 model is strikingly similar with rat, G13 and other bat coronaviruses. Now RBD of RM&Y2, the bat coronavirus RATG13 and two pangolin beta coronaviruses using homology modeling was analyzed. Two amino acid deletions in RM&Y2 RBD made two loops near the receptor binding site that are shorter than those in SARS-CoV-2 RBD. Deletions in RM&Y2 reduces the binding with ACE2 or even cause non-binding. Now in the figure we can see the figure B, RATG30, the figure C, pangolin MP789 2019, and the figure D, pangolin P5L 2017 does not have deletions, and similar conformation indicates that they may also use ACE2 as cell receptor. Pangolin MP789 2019 shows higher homology with SARS-CoV-2. Now SARS-CoV-2 shows homology with bat and pangolin coronaviruses. So are pangolins the intermediate host? 5 key amino acids in RBD are consistent between pangolin CoV and SARS-CoV-2. Aside from RATG13 and pangolin CoV, aside from RATG13 and other rat coronaviruses, pangolin CoVs are the most closely related CoV to SARS-CoV-2. S1 protein of pangolin CoV is much closely related to SARS-CoV-2 than the RATG13. 5 key amino acid residues are completely consistent, as we can see here, between pangolin CoV and SARS-CoV-2. However, 4 amino acid mutations are also present in in RATG13. Both pangolin CoV and RATG13 lost the putative furin recognition sequence motif at S1 S2 cleavage site that can be observed in SARS-CoV-2. Conclusively, studies suggest that pangolin species are natural reservoir of SARS-CoV-2 like viruses. Now, Malayan pangolin CoV and SARS-CoV-2 is a phylogenetic relationship which we can see clearly here, suggesting pangolin as the intermediate host. As we can see, there are many common areas in SARS-CoV-2, pangolin CoV, and RATG13. Pangolin CoV genome consists of 6 open ORFs, open-ending frame, common to CoVs of RATG13 and SARS-CoV-2. Some pangolin CoV shows higher amino acid sequence identity to SARS-CoV-2 genes than to RATG13 genes, in some cases such as ORF1b, S protein, ORF7a, and ORF10. The high spike protein amino acid identity implies functional similarity between pangolin CoVs and SARS-CoV-2. Considering the wide spread of SARS-CoV in natural reservoirs such as bats, camels, pangolins, SARS-CoV-2 may have transmitted from bats to pangolins and then ultimately unrestricted human-to-human transmission. So, as far to conclude, RATG13 and RMNY2 and some other pangolin coronaviruses shows the much homology with SARS-CoV-2. Similarity of RBD with other coronaviruses with some additions or deletions suggests that the virus may have originated via mutations and recombinations during adaptations. Bats and pangolins must have played the important role in undetected zoonotic transmission of SARS-CoV-2. Therefore, it can be safely concluded that SARS-CoV-2 is a result of natural selection and not product of genetic engineering by PRC. I would like to express my gratitude to the organizing committee and my professor, Sri Biswajit Saha. I'd also like to express my special thanks of gratitude to my friends and my family for the constant support and motivation. I have took several references which are mentioned in this slide. Thank you. Till then, stay safe. This shall overcome.
Hello.
Yes, ma'am.
Yes. Thanks. Thanks for your presentation. মালবিকাও আছে, মালবিকা কিছু কোয়েশ্চেন করবে? হ্যালো, কিছু বলবে? চিন্ময়?
ঠিক আছে, অসুবিধা নেই।
না, ভালোই
ঠিক আছে,
হয়েছে।
ভালোই হয়েছে।
আচ্ছা, নেক্সট হচ্ছে দীপান্বিতা শেঠ, মাইক্রোবায়োলজি, সুরেন্দ্রনাথ কলেজ। Dipannita, are you here?
Not present. Even she will not present. She is absent.
Dipannita said absent.
Yes.
Dipannita Seth absent. Dipannita C.
Ma'am, even she will not present. She is absent too.
Oh, both are absent.
Snigdha Mukherjee, microbiology, Vijay Krishna Girls College. Snigdha?
Yes, ma'am, present.
Okay. Welcome, beta. Your topic is a review on microbial biodiversity in glacier ecosystem.
Yes, ma'am.
Yes. Carry on, beta. Start. Screen share karo.
হ্যাঁ ম্যাম করেছি। Ma'am, is it visible?
Yes, it is visible. Nicely.
And mind on the time restrictions, please.
Yes. Now it is 7:34. 8 minutes you have to do. Just start.
Good evening, everyone. I'm Snigdha Mukherjee, 5th semester BSc Honours student of Department of Microbiology of Vijay Krishna Girls College. My topic of presentation is microbial biodiversity in glacier ecosystem. Let me first describe my topic in the introduction. As we all know that microbial biodiversity is a variety of range of different kinds of unicellular organisms like bacteria, archaea, protists, and fungi. Glaciers, on the other hand, are formed due to harsh environmental conditions like low temperature, high UV radiation, and limited number of nutrients. Researchers have found some photosynthetic microorganisms on the glacial surface like snow algae and cyanobacteria, thus making an ecosystem on glaciers. Now we are going to see the various types of glacial ecosystem. The top surface of glacier is receiving solar radiation, atmospheric depression, and precipitation also, forming the supraglacial zone. Just beneath that lies the endglacial zone. At last lies the subglacial zone, which is getting all the chemical energy, rock comminution, and all groundwater resources. Proglacial streams serves as a link between the glacial surface and the groundwater resources, and getting mixed into rivers, lakes, oceans, and marine habitats. Now we'll see in details of these various zones. The supraglacial ecosystem comprises of the top layer of ice, so it receives all the atmospheric gradient. And largest supraglacial ecosystem is Greenland ice sheet. If we move to the biological community, then it comprises of the autotrophic and heterotrophic bacteria. For example, Spingobacterial species and Pseudoanabana species, etc. are found in this ecosystem. Next is the endglacial ecosystem. It is found in the interior of the glacier where light does not penetrate, so the area has a high amount of pressure and a nearly absence of water. When the surface melts, the fluid passes through this endglacial zone, and there a microbial community grows. Planococcus, Halocryophilus, and other similar species are found here. Next is the subglacial lakes, which are found under the glacier, beneath the ice sheet. Subglacial lakes contain about 15% of all liquid fresh water on Earth. Some extremophilic microbial community has also been found here, such as Marinobacter species. Next is the proglacial streams. So proglacial streams serve as a link between glacial processes and a downstream habitat, both freshwater and marine. For this ecosystem, focal species are macroinvertebrates. Here is a picture of Lake Tasman, which is more than 200 meters deep, and proglacial ecosystem has been found here. And in the picture, we can see how the icebergs have formed and how the moraine has been thinned at some portion, and little ice edge lateral moraine are also seen here. Next is the glacial forefield. It is the region between the edge of the glacier and the moraines of Lepus maximum. Bacterial community is similar to supra and subglacial habitat. fungi and bacteria play an important role in primary succession of glacial foreland. Next is the environmental factors which are adversely affecting the glacial ecosystem. As we all know that global warming, rising sea levels and carbon emissions, these are the most affecting factors. We can see that how global warming leading to melting of snow in polar areas causing a threat to the polar creatures there, and also rising sea level causing a huge damage to the coastal areas and the people living there. And at last, this graph represents India's carbon dioxide emissions fall for the first time since 1982, which has happened in this 2020. This happened due to the lockdown for COVID-19 and all public and private sector were kept closed, so the pollution level came down drastically. Now we go to research work. Many scientists have researched on this topic. Linda Wellham on March 2013 used 26 alpine glaciers and by method of 454 pyrosequencing of the 16S rRNA genes found that the biofilm community composition. Mostly proteobacteria, bacteroidetes, actinobacteria and cyanobacteria are found in this glacier ecosystem. We move to the future prospects. It is estimated that by 2050 about only a quarter of today's glacier will exist. So it is a big threat and there won't be any glaciers below this altitude either, as snow is necessary for the glaciers to survive. At last, I'm concluding my slide. We must try to preserve the glaciers as it has a profound influence on the global environment. These are my references, and I would like to thank our principal ma'am, Dr. Rupa Bhattacharya of Vijay Krishna Girls College, and all the organizers for giving us such a platform to deliver this topic of my interest. And I'm very much thankful to Shri Vishwajit Shaha, Faculty of Department of Microbiology of Vijay Krishna Girls College for his extreme guidance. And thank you everyone for your kind attention. Thank you all.
Thank you, Snigdha.
Thank you.
Chinmoy?
It's okay. Actually, Snigdha has mentioned the topic, a review on the microbial biodiversity in the glacial ecosystem, and he should give a touch on the writings like the threats to the microbial biodiversity.
হ্যাঁ, যাই হোক তবুও ওরা এইটা করেছে, হ্যাঁ ভালো হয়েছে। And thanks to your teacher also.
Vishwajit is here also. Vishwajit, you want to ask any question?
ঠিক আছে, ভালো প্রেজেন্টেশন করেছে, স্লোলি খুব সুন্দর করে
ভালো প্রেজেন্টেশন করেছে। হ্যাঁ, এই যে অভ্যাসটা হচ্ছে এটি একটা বড় কথা। খুব ভালো হয়েছে।
Thank you, Snigdha.
Thank you, Snigdha. Next, Abhilasha Ganguly.
Yes, ma'am. Am I
Anandaram Joypuria College.
Yes, you are audible.
Abhilasha, yes.
Thank you,
Your
ma'am.
topic is mangrove forest and sustainability of Sundarban Delta, isn't it?
Thank you, ma'am, for giving us the opportunity. Yes, you are right. That is my topic. I want to request my fellow Goirika Bhattacharya to present the screen.
Oh, screen share karo, Mallika.
Goirika Bhattacharya.
Is it visible, ma'am?
Okay. Yes, it is visible.
Thank you, Goirika. Good evening to all. I am Abhilasha Ganguly, same 3 students of St. Anandaram Joypuria College from Department of Zoology. Today, I and my fellow Goirika Bhattacharya will highlight on the topic mangrove forest and sustainability of Sundarban Delta. Mangroves, the only tall forest on earth where land and freshwater and saltwater mix are uniquely adapted to tropical and subtropical coast as they cannot withstand freezing temperature. Here in the slide you can see Bengali name Sundarban means beautiful forest that have been derived from the word Sundri or Sundari, the local name of the mangrove species. The delta is a mangrove area formed by the Ganga, Brahmaputra and Meghna river in the Bay of Bengal. It spans from the Hooghly river of West Bengal to the Baleshwar river of Bangladesh. This is one of the largest such forests in the world, occupying about 10,000 square kilometers across India and Bangladesh, of which 40% lies in India. Here you will find halophytes, the salt-tolerant plants. But this delta is ecologically fragile and climatically vulnerable because it faces frequent cyclone, heavy rain, storm and flood that causes land erosion. They faces low scope of cultivation and low scope for artificial culture for frequent storm and salinity of soil. Last but not the least, they faces loss of biodiversity and unstable life and livelihood of people living there. Now, I will ask my fellow Goirika Bhattacharya for further continuation.
Good evening to all. I shall mainly explain to you about mangrove forest and its role in sustainability of Sundarban Delta. And I shall mainly focus on sustainability of soil, environment, biodiversity, and sustainability of life and livelihood of people. Now I would like to seek your attention on sustainability of soil at first. The soil stratum of the delta is in dynamic construction by the characteristic root systems of mangroves. The main cable root system of the mangrove trees, comprising of aerial pneumatophores and underground anchoring roots, slows down the movement of tidal waters that results in the sediments to settle and build up the muddy bottom. Not only that, almost all mangroves are shallow rooted and some are deep rooted or have persistent tap roots, but the root systems are often extensive and may cover a wide area. The intricate roots of mangroves makes these forests attractive to fish and other organisms for seeking food and shelter. Now I will come to sustainability of environment. Mangroves can absorb different greenhouse gases like methane, carbon dioxide, etc. That's why they are called natural carbon sinks, and this carbon is mainly called blue carbon. It reduces the warming of Earth. A study financed by the Union government and headed by noted marine scientist Abhijit Mitra estimated that in the central part of Sundarban near Matla River, the capability of pine trees to absorb carbon was 22 tons per hectare. But in the eastern Sundarbans, the capacity of pine to absorb carbons is near about 35 tons per hectare. Now we should also know about the sustainability of biodiversity. And in this slide, you can vividly understand that Sundarban's forest is one of the richest biodiversity hotspots in India, containing about 320 birds, 400 fish, 49 mammals, etc. And home for many rare, globally threatened wildlife species like Royal Bengal tiger, Gangetic dolphin, olive redleg turtle, etc. On the other hand, sustainability of livelihood of man is a great deal too. Sundarban has over 4.5 million population, and according to the birth chart, it increases gradually, which largely depends on the mangrove forest, ranging from collection of honey, timber, herbs as food, boat, and Ayurvedic medicines, to the culture of fishes and prawns in the ponds built within the parts of mangrove forest. They have used different mangrove plants in different forms as medicine to cure some chronic ailments like fever, malaria, leprosy, etc. And in our reviews, we have tried to document the medicinal uses of Sundarban's mangrove plants by collecting data from local people and Ayurvedic practitioners. Altogether, 35 mangrove species, including 23 trees, 7 shrubs, 3 herbs, 1 semi-deciduous tree, and 1 woody climbing shrub under 20 botanical families, which are used to cure ailments commonly by the local healers. Sundarban mangrove are in great vulnerable condition nowadays because they are decreasing gradually every year, and the reasons are already explained by my co-presenter Abhirasha Ganguly. Now I will come to our conclusion, mangrove as gift to us. Mangrove forest provides coastal protection through surge and wind suppression during tropical cyclones and often recover with time and continue to provide numerous societal benefits in the wake of storm events. Mangroves provide essential habitats for thousands of species and also stabilize shorelines, protect the lands by preventing erosion and people from waves and storms. But the mangrove forest is very sensitive to pollution and declining at the rate of 1% per year due to the climate change and cyclones. Therefore, the sustainability of Sundarban Delta requires a long-term vision that can integrate climate adaptation and strategies for conservation of mangrove forest and its biodiversity. We got our references from these sites. We do acknowledge Vijay Krishna Girls College at first for this beautiful initiative and for providing us with this great opportunity to embark on this project. We also wish to show our gratitude to Dr. Ipshita Chanda ma'am, Assistant Professor and other faculty members of Department of Zoology, St. Anandram Joypuria College, Kolkata, for their sincere help and guidance. Thank you so much for your patience and attention. Keep your world colorful and preserve biodiversity. Thank you.
Thank you, Abhilasha, for your presentation, and thanks to your teachers also. Very nice presentation. Chinmoy,
Yes,
or others.
I must give thanks to Abhilasha as well as Goirika also, because
Yes, Goirika also.
For their cooperation and the coordination. Nicely they have presented their preparation. Very good. Both of you.
Thank you, sir. Thank you, sir.
And topic is very burning topic.
Very good.
Now, Shaptadipa Mukherjee, Jogomaya Devi College.
Yes, ma'am.
Shaptadipa,
Yes,
are you
ma'am.
here?
Yes,
Okay.
ma'am.
Start. Your topic is survival of the
monarch butterfly.
Monarch butterflies. Oh, very
Yes,
good. Okay. Start, beta.
Ma'am, is it visible?
Yes, visible.
Yes, it is visible.
Okay. Respected teachers and my fellow students, myself Shaptadipa Mukherjee from Jogomaya Devi College 3rd year would like to present to you about the survival of the monarch butterflies. This is a study of the monarchs, their fitness and survival in this adverse climatic condition, and also how the host plant of the beautiful butterflies, the milkweeds, can turn out to be fatal to the butterflies. These are the points that I would like to talk about and share with you different research results and explanation of different scientists. The monarchs are one of the most beautiful butterflies in the world, which are native to the North and South America. Their breeding places and migratory pathway has been traced by several scientists. Here in the picture, we can see the beautiful male and female monarchs and their distribution of the butterflies. Climate change and the host plant milkweed has been the major reason for several of years for the rapid decrease of the monarchs. We all know that the climate across the globe is undergoing tremendous change. All the living organisms are being affected and the monarchs are no exception. Here in the graph of the monarch population of Mexico, we can see very well that the population has been decreasing for almost a decade. The milkweeds also become fatal for the monarchs, and we will know why in the later slides. There are several cases of climate change which affects the monarch population. I have put up one of the many. Monarch butterflies are bioindicators, and climate change triggered them to show their migratory response. But due to the increase in temperature in Canada, they failed to travel south. By the time the temperature was suitable enough to trigger their migratory instincts, the climate in Midwest is so cold that half of them die during their migratory journey. An instance happened in 2002. A sudden storm killed 80% of the overwintering monarch population in Mexico. Again, in hotter and drier weather conditions have also been proven to be lethal for the monarch butterflies. We have seen how the climate change has been proved to be lethal for the monarch population. Now the question arises that the milkweed, which is the host plant of the monarchs, are they safe? Do they help wholly in the survival of the monarchs? The answer is no. Here I have listed two types of milkweeds, non-native and native, and few of their characteristics. The most important point to be noted is that the non-native or the exotic milkweed does not dormant in the winter like the native milkweeds, and thus it caused several non-migratory groups to form on it. Studies show that the butterflies which breed on the non-native milkweed has a higher reproductive success, but the problem arises when the migratory monarchs form non-migratory groups on the year-long growing exotic milkweeds. The milkweeds host a protozoan parasite which causes infection in the caterpillars which breed on the milkweed. In case of native milkweeds, the protozoans die along with the plant, but in non-native milkweeds, it is not the same case. The non-native milkweeds does not dormant. Thus, the protozoan infection spreads more and more among the non-migratory monarch butterflies. Here is an experiment worked out by scientists. An open-top chamber or OTC was taken and few monarch butterflies were left with both native and non-native milkweeds and observed for several weeks. Temperature was increased to detect the survival, adult mass, forewing length of the butterflies in the elevated temperature. It was seen that the monarch survived more at first in the elevated temperature feeding on A. incarnata, that is the exotic milkweed, than on the native milkweed. But in a more elevated condition, the monarch started dying as the plant started releasing a toxic chemical called cardinolite. Increased temperature also reduced the forewing length. Thus, the cardinolite concentration transitioned from beneficial to detrimental. Hence, it was proved how temperature change and milkweeds can be fatal for the monarch butterflies. Few measures have already been started to be taken. In US, massive efforts are being done for saving the butterflies. More awareness should be spread in order to keep the climate change in check. Hence, it is to conclude that both climate and the host plants are to be taken care of for saving the iconic and gorgeous monarch butterflies. Thank you. This is Shoptadipa Mukherjee.
Thank you. Thank you, Shoptadipa.
Nice presentation.
Very nice.
Thank you, sir.
Topic also.
Thank you.
And your pictures are also very nice.
Thank you, ma'am.
Chinmoy, কিছু বলবে?
Oh, better to call next. পরে ভালো হয়েছে তো, সুন্দর হয়েছে, সুন্দর বলেছে, নাইস প্রেজেন্টেশন।
থ্যাংক ইউ স্যার।
আর কারো কিছু জিজ্ঞেস করেনি তো? আচ্ছা, একবার সুশান্ত, আমরা তো শেষের দিকে এগোচ্ছি, একবার যাদের ডেকেছিলাম প্রথম দিকে আসেনি, একবার দেখে নাও তারা কেউ জয়েন করেছে কিনা। আমি এর পরের জনকে ডাকছি। পরের জন হচ্ছে সৃজনী সেন, বিবেকানন্দ কলেজ। সৃজনী, are you here, beta?
Yes, ma'am.
Oh, okay. Start. Your topic is braving the odds for biodiversity. বাহ, শুরু করো বাবু। হ্যাঁ, সুশান্ত।
হ্যাঁ ম্যাডাম।
অমৃতা, একবার দেখে নাও যাদের আমরা আগে ডেকেছি কেউ এসেছেন কিনা, বা কোনো মানে তাদের নেট প্রবলেম ছিল, এখন জয়েন করতে পেরেছে কিনা।
আমি চেক করে নিচ্ছি। ও বলুন,
ইতিমধ্যে ও শুরু করুন। সৃজনী, বেটা স্টার্ট।
Yes, ma'am. Ma'am, is it visible?
Yes, nicely.
Yes.
Okay. A very good evening to one and all. On the onset of the event, I would like to thank the organizers of the 2-day international webinar organized by IQAC Bijoy Krishna Girls College for giving me the opportunity to present my work. I am Srijoni Sen, 4th semester undergraduate student from the Department of Zoology, Vivekananda College, Thakurpukur, to give a generalized account on biodiversity and associated sustainable means. Biodiversity forms the basis of global life support system, providing a variety of environmental services that are essential at the global, regional, and local level. Man heavily depends on natural ecosystems for essential ecological services like maintenance of atmosphere, regulating weather patterns, pollination of crops,
water cycle, maintenance of genetic library, etc. Sustainable development, on the other hand, is economic development that is conducted without the depletion of natural resources, so that the future generations are not left empty-handed. Biodiversity development is social plus economic development. Equitable development is social plus economic development. And viable development is environmental plus economic progress. The United Nations has published 17 Sustainable Development Goals to guide the role of public, non-profit and voluntary sector in sustainable development. Notable amongst them are end poverty in all forms of life, end hunger, achieve food security and improve nourishment, ensuring inclusive and equitable quality of education, achieving gender equality and empowerment of women and girls especially, reducing inequality amongst the nations, and strengthen the means of implementation and revitalize the global partnership. The Paris Agreement under the United Nations Framework Convention on Climate Change, also called the Paris Climate Agreement or COP21, is an international treaty adopted in December 2015. The main objective was holding the increase in the global average temperature to well below 2 degrees Celsius above the pre-industrial levels, and to pursue efforts to limit the temperature increase to 1.5 degrees Celsius above the pre-industrial level. Enrichment of industries due to the sudden spike of single-use plastic commodities as per the National Oceanic and Atmospheric Administration, abbreviated as NOAA, in COVID-19 times has urged the high-level political forums within its Global Partnership on Marine Litter, abbreviated as GPML, to take remedial measures for healthier oceans. Artificial intelligence can also integrate data, and several multi-sector collaboration platforms can also share information to generate the awareness. Ecosystem services are the gifts which we get from nature, and these are services which can be of four types: provisioning, supporting, regulating, and cultural. It is necessary to be responsive, adaptive, and innovative to achieve the best fit within the system's dynamic environment. Because the ecosystem services upholds the central value of respect and nurture of the essence and uniqueness of a healthy and resilient community. The ecological footprint is a method which is promoted by the Global Footprint Network to measure the human demand on natural capital, that is, the quantity of nature it takes to support people or an economy of a country. The Global Footprint Network estimates as of 2016 that humanity has been using natural capital 1.7 times faster than the Earth can renew it, which they describe as humanity's ecological footprint corresponds to 1.7 planet Earth. This leads to a condition called overshoot, as you can see demarcated the red portion in the graph. We humans are in the middle of a big extinction crisis right now. This picture clearly shows that extinction of one particular species, even the smallest one, will affect our survival and existence drastically. Because we have already seen that recently 801 species are totally wiped out in the last decade, and 10,104 vulnerable species are newly enlisted. So this picture clearly shows our interconnection with the diverse species and ecosystems. During the pandemic, we have seen the re-emergence of various important species. Notable amongst them are the green sea turtle, that is the Chelonia midas, the rainbow toad, which is the last picture, and most importantly, the birth of 3 baby chicks in Sussex, England, of the species Syconia syconia, which is a record registered after 604 years in May 2020. Talking about the Indian perspective, the Ramsar site is a wetland site designated to be of international importance under the Ramsar Convention. The prime objective of conservation will be to analyze the impact of the loss of wetlands not only on the ecology but also on the economy of a country, to devise management strategies of the government organization, and to suggest means for the conservation of this fragile ecosystem. The present research work has been based on intensive field study, sample survey, collection of primary data, and analysis using GIS and remote sensing technology. Next, I come to a term that is corporate social responsibility, which is now accepted as a means to achieve sustainable development of an organization. It is about integrating economic, environmental, and social objectives with the company's operation and growth. The Companies Act of 2013 will make Indian companies spend 2% of their profit on these CSR activities. Sundarbans is a mangrove forest located in the coastal region and is also a UNESCO World Heritage Site. Currently, mangrove forests in Sundarbans are facing destruction due to anthropogenic causes like pollution growth, shrimp cultivation, timber and raw material collection, waterlogging, especially after Super Cyclone Amphan had hit us hard on 28 May 2020. So every topic discussed in the global section contributes to a loss of biodiversity in these mangroves. And these include several endangered species such as the Irrawaddy dolphin, the Indian python, and the estuarine crocodile. Virtually all the flora and fauna in the Sundarban is being now threatened by global climate change. And for the last 40 years, the discharge of sediment-laden freshwater into the Bay of Bengal through the Bangladesh portion has created several disruptions to our mangroves. And hence, a mutual collaboration between countries like India, Bangladesh, and neighboring nations is of urgent importance. Finally, I would like to conclude with what Sir Thomas Eisner, an American-German entomologist and ecologist, also regarded as the father of chemical ecology, beautifully said that biodiversity is the greatest treasure we have, and it is our responsibility to nurture and protect our nature, our mother earth. And last but not the least, I would like to express my heartfelt gratitude to Dr. Malavika Bhattacharjee, HOD and Assistant Professor, Postgraduate Department of Zoology, for her immense support and thorough guidance to help me prepare for the presentation and today's talk. Thank you everyone for your kind attention. Thank you.
Thank you. Thank you, Srijani.
Thank you, ma'am.
Chinmoy?
It's okay. Very good. Thank you.
Very good.
And
Very
thank
nice presentation.
Topic
Now,
is very
the next one, Priyanka De of Sarshuna College. Priyanka? Hello, Priyanka, are you here? Priyanka absent? Amrita?
No, ma'am, Priyanka is not present here, I think.
Oh, okay.
And I have gone through the participant list. Those who were absent previously yet not have joined. So we can mark them as absent.
Okay. Next person, Kothon Bandyopadhyay, I think he has presented.
Yes, presented. Right.
Okay. Next, Himadri Chakraborty and Ishani Chaudhuri, Surendranath College. Himadri or Ishani, are you present?
No, madam, ওরা নেই।
আচ্ছা।
ওরা attendance list এ নেই।
আচ্ছা, সায়ন নায়েক, সরসুনা কলেজ। সায়ন?
Loss of biodiversity.
Shayan is also absent.
সায়নও নেই।
আচ্ছা, নেক্সট হচ্ছে সুদত্তা Vijay Krishna Girls College. Yes. Sudatta. Your topic is Panthera tigris tigris versus Homo sapiens. Yes, man-animal conflict. Very good. Peace, conflict, or extinction. A review. Very good topic. Sudatta, start. Start, beta.
Yes, ma'am. Is it visible?
No, beta, screen share karo. Yes, it is visible. Yes, now it is visible.
Okay, thank you, ma'am. I am going to present on Panthera tigris tigris versus Homo sapiens, peace, conflict, or extinction. Let's see the Bengal to the current status. Since 2006, science-based tiger census has been conducted before year to determine their number and location. According to the graph, we see the hike in tiger population from the year 2006 to 2018, the number has risen from 1,411 to 2,967. This frequency depicts the height in the tiger's population. Now, characteristic of Bengal tiger. It has icy blue eyes. Here is the scientific classification of Bengal. Here are some magnificent pictorial of Royal Bengal tiger. Now, habit and range of Bengal tiger. The habit of Bengal tiger real range of Bengal tiger from Western Russia, mainly Bangladesh, Bhutan, India, Indonesia, Myanmar, Nepal, Russia, Thailand, etc. This map depicts distribution of Bengal tiger in Sundarban Tiger Reserve, India. This depicts historic and distribution of Bengal tiger in the world. Now, case of Bengal tiger. According to IUCN, the Bengal tiger is in danger of extinction. Most species, its number keep falling dramatically over the past centuries rather than increasing. Habit loss, the extreme large-scale incidents of poaching and conflict with humans over areas are serious threats to the species following the first. Three is the loss of habits of due to agriculture and industrial treatment, timber cutting, new roads, etc. Tiger's habit, that is the forest, mountains, and grasslands, are fragmented, grazing in the habitation of wilderness has come up with the tigers of survival. Next is increasing Indian population. Asia's explosive population growth demands that more and more land be converted to agriculture. In India, where about 60% of the world wild tigers still roam, human population has grown by 50% in the past 20 years. Next, human-tiger conflict. The floating habit where tigers have activities is also one which has housed one of the most concentrated and rapidly expanding human population. At the beginning of the 19th century, tigers are so numerous and seems to be as to whether man or tiger would survive. It became the official policy. The killing of tigers will reward, being paid for destruction in many localities. Tigers in the Sundarban presumably attack humans who enter their territories in search of wood, food, honey, or they find next competition. As tigers for land, they find less and less to eat. Local people hunt the same prey of as tigers do, pressing tigers to resort the domestic animal and one rare occasions when humans. Predators often shoot or snap the encroaching tiger. Last but not the least, the most significant immediate threat to the extinction of tiger population is the illegal trade in poaching skin and bone part from China. The demand for bones and body parts from wild tigers for use in traditional medicine in the reason for the unrelenting poaching pressure on tigers on the Indian subcontinent. Here is some pictures of tigers' bones and skins. Conservation of Bengal tigers. An area of special interest lies in landscape in the ruthless of northern India and southern. WA from a global region saved tigers now with
goal of building political, financial, and public support to double the wild tiger population by 2022. Save Tigers now started its competition in 12 different WWF Tiger steps 2010. In 1972, Project Tiger was launched, aiming at ensuring a viable fish of tigers in the country as preserving areas of biological importance. The Indian Wildlife Protection Act of 1972 enables government agents to take strict measures as to ensure the conservation of the Bengal tigers. There is still hope. We can save wild tigers. We are concentrating our effort on protecting key landscape where the big cats have the best chance of increasing for the long term. 5 decades of conservation experience has enough space, prey and protection to recover. By saving also the biological and diverse landscape where they still roam, Indians large rainforests, jungles, and birds. These forests are home to thousands of other species. People and the food, fresh water, and flood protection that local communities need to survive. We should tigers because number 1, tigers are admired by many people. Number 2, a home for tigers means a home for others. Number 3, population healthy ecosystem. Number 4, tigers have a positive effect of communities. Tigers are strong and determined. These are some of the references I have used for preparing my presentation. Put to my heart gratitude to and USC coordinator, Ma'am Dr. Presetatio, for giving me this opportunity to topic in this platform. I would also like to express my gratitude to our HOD, Dr. Papia, my other to of support. Thank you all for your patience.
থ্যাংক ইউ সুদত্তা। আমরা আশাবাদী, তোমার সাথে সাথে আমরাও আশা করছি আমরা এই সমস্যা কাটিয়ে উঠবো। উইল ওভারকাম দ্য প্রবলেম। চিন্ময় বা মালবিকা, মালবিকা আছিস? প্রশ্ন করবি কিছু?
ম্যাডাম আছেন?
আছি আছি আছি।
কিছু বলবি? একটু বল না।
না, ওরা দেখো প্রথমবার অনেকে প্রেজেন্ট করছে, তো আমার তো ভীষণই ভালো লাগছে না।
আমার তো ভীষণ ভালো লাগছে।
এবং হয়তো কিছু কিছু কেউ নার্ভাস হয়ে, যেমন 1-2 জন সামনের দিকে তাড়াহুড়ো করে ফেলেছিল বা পাওয়ার পয়েন্টকে প্রপারলি প্রেজেন্ট করতে পারছিল না। বাট আদারওয়াইজ মানে আমি নিজে ভাবছি যে আমাকে যদি আমার টাইম এরকম প্রেজেন্ট করতে বলা হতো, আমি তো ঘাবড়ে শেষ করতাম। সেই দিক দিয়ে ওরা ভীষণ স্মার্টলি প্রেজেন্ট করে যাচ্ছে, ওয়ান আফটার অ্যানাদার।
আমি প্রশ্ন কেন করতে বলছি, প্রথম দিকে সবাইকে প্রশ্নটা করিনি টাইমের জন্য। এখন অনেকে আসেনি বলে টাইম আছে। একটু প্রশ্ন ফেস করতে শিখো, আর কিছু না। হ্যাঁ, একটু প্রশ্ন করা ভালো। প্রশ্ন ফেস করতে শিখলে না স্মার্টনেসটা খুব ভালো বাড়ে, বুঝেছিস? হ্যাঁ, একটুখানি দেখ, যদি কিছু প্রশ্ন করিস। খুব ভালো হয়েছে সুদত্তা। হ্যাঁ, জানো তো, যে বনে বাঘ থাকে, যে বনে বাঘ থাকে সে বনের সমস্ত প্রাণী খুব সুখে থাকে। কারণ বাঘের মানে প্রাণীদের একমাত্র শত্রু, বনের শত্রু হচ্ছে আমরা মানুষ।
তুমি এটা বললে বলে আই এম আস্কিং আ কোয়েশ্চেন। আমব্রেলা স্পিসিস আর কিস্টোন স্পিসিস জানো? টাইগারকে বলা হয় কিস্টোন স্পিসিস। আইডিয়া আছে কেন?
নো ম্যাম।
সুদত্তা
না ম্যাম,
পড়োনি না ওটা?
না।
আচ্ছা, আসলে ওরা আছে বলে আমরা বেঁচে আছি, জানো? সমস্ত বনটা বেঁচে আছে ওই ছাতার তলায়, ওই বাঘের জন্য। ওই যে বললাম, যে বনে বাঘ থাকে সব প্রাণী খুব সুখে থাকে। খুব ভালো প্রশ্ন হয়েছে। আর কিছু? ওইটা ও পড়েনি তো মালবিকা?
হ্যাঁ, তাই তো দেখছি না। যাই হোক, সেটা পড়েনি, ঠিক আছে।
হ্যাঁ হ্যাঁ, ওটা
তুমি নিজে দেখেছো কোথাও বাঘ সামনাসামনি দেখেছো? মানে হ্যাভ ইউ সিন ইন ইওর রিয়েল লাইফ? মানে সাফারিতে গিয়ে দেখেছো?
না না ম্যাম, সাফারি গিয়ে দেখিনি।
আচ্ছা, ওটা জুলজিক্যাল গার্ডেনে দেখেছো? সুদত্তা? আচ্ছা।
জুলজিক্যাল গার্ডেনে দেখলে ওইটা বুঝতে পারবে না, বিকজ দে মুভ ভেরি সাইলেন্টলি তো, আর এত ভালো ক্যামোফ্লেজ করে। এইটা একটু জঙ্গলে না গিয়ে দেখলে মৈত্রী দিয়ে ওরা বুঝতে পারবে না।
একদম একদম একদম।
যাক, ঠিক আছে, খুব ভালো বলেছে।
ভালো বলেছে, সবাই খুব ভালো বলেছে, প্রত্যেকের প্রেজেন্টেশন ভালো হয়েছে। হ্যাঁ হ্যাঁ, এবং থ্যাঙ্কস টু অল দ্য টিচার্স, দোজ হু হ্যাজ গাইডেড। প্লিজ, মানে আমি খুব থ্যাঙ্কফুল ফর দ্য, মানে টু দ্য টিচার্স। খুব ভালো লাগলো। আচ্ছা, অমৃতা, হ্যাঁ এবার আচ্ছা, কিংবা আর তো কেউ নেই বাবু?
আর কেউ নেই, একটুখানি আর আমাদের
এবার তোমরা শেষ করো তোমাদের যা যা করণীয় গুলো।
হ্যাঁ, আমি দেখছি।
আজকে ভোট অফ থ্যাঙ্কস দেখো কি আছে।
হ্যালো ম্যাম, অ্যাকচুয়ালি একজন পরে জয়েন করেছে, যার নেটওয়ার্কের জন্য জয়েন করতে পারেনি। কলেজ।
ও, ভেরি গুড। বলো। দীপান্বিতা?
Yes, ma'am.
Yes.
You start.
Your topic is, হ্যাঁ শিওরলি। আমি তো অনেকক্ষণ থেকে খুঁজছি যে কে পারলো না। দীপান্বিতার টপিক হচ্ছে আন্ডারস্ট্যান্ডিং দ্য রোল অফ হিউম্যান microbiota in human health. Yes,
right.
হ্যাঁ, দীপান্বিতা, স্টার্ট বেটা।
Ma'am, I'm presenting my screen. Yes.
Dipannita, it is visible. Yes, start.
Okay, myself, Dipannita Shi, Department of Microbiology, THK Jain College, Kolkata. My topic is understanding the human microbiota in human health. Introduction. Human body are constantly in contact with the nature all the time. However, we are in even more initiate contact with an enormous number of microorganisms that inhabit our bodies. The microbiome refers to the collective genomes of the microorganisms in the particular environment, and the term microbiota is the community of microorganisms in a particular environment, and microbiota is the community of microorganisms themselves. Approximately 100 trillion microorganisms like bacteria, virus, protozoa, algae, fungi are present in our body. The normal microflora are more or less contact with each species and are broadly divided into resident and transient microflora. This slide tells about many species of microorganisms present in our body. This slide is the type of microflora is normally divided into transient microflora and resident microflora, and third, carrier state. Resident microflora present in our body for a long time, and transient microflora present for a while in our body. Carrier state, this type is different from potential pathogens. Resident microflora, it constitute a constant population which cannot be completely removed permanently. They resident prevent permanent colonization of the body by other microorganisms. Example is Staphylococcus epidermis is the dominant species of resident microflora. Resident microflora are normally present in our waves, scalp, normally our moisture areas. This slide is about transient microflora. May be potentially pathogenic or non-pathogenic microorganisms that are present for some times, not for all the times. Example, Streptococcus pyogenes.
in throat. They cannot persist in our body because of the competition with the other microorganisms. The sterile microorganisms in human body. A healthy human body's sterile parts are normally blood, cerebrospinal fluid, perineal fluid, including chest fluid, bone marrow, bone joint fluid, including synovial fluid, etc. Sterilization maintained by local defense mechanism and chemical substance in the serum and tissues. Examples, antibodies. This slide is about the normal microflora present in our body. First, our skin. Second, the oral cavity. Third, respiratory tract. Fourth, gut intestinal tract, genetical tract, etc. Normal microflora in our skin. An average adult healthy human body is covered with approximately 2 to the power m square of the skin. The surface area is about 10 to the power 12. Most skin microorganisms are associated with the apocrine gland, which is sweat gland. Each hair follicle is associated with sebaceous gland, which secretes the lubricant fluid. The responsible microorganisms are Acinetobacter, Enterobacter, Klebsiella, etc. Normal microflora of the oral cavity. Our oral cavity is full of microorganisms, we all know. The normal microflora of the oral cavity constitute of many kinds of microorganisms like Streptococcus, Lactobacillus, Neisseria, and some fungi also, etc. The microorganisms are colonized in the cavity due to the comfortable environment. Normal microflora of respiratory tract. The respiratory tract is divided into two, upper respiratory tract and lower respiratory tract. The upper respiratory tract is normally found Streptococcus, Staphylococcus, and Neisseria, etc. And the lower respiratory tract has no microflora because of the upper respiratory have the most of the microorganisms, so the microorganisms cannot reach in our lower respiratory tract. Normal microflora of gut microbiota. The human gastrointestinal tract, the food of digestion, consists of stomach, small intestine, and large intestine. Stomach. Many microorganisms are washed from our oral cavity to stomach, but most of them are killed by stomach fluid because of acidic pH. Example, Helicobacter, Streptococcus, Staphylococcus, and Lactobacillus, etc. Small intestine. The small intestine divided into three parts: duodenum, jejunum, ileum. Duodenum attached with the stomach, so it is acidic in nature. So gram-positive cocci and bacilli are present here. Jejunum. It contains lactobacillus, diphtheroid, Enterococcus faecalis, and Candida albicans. Ileum. The distal part of intestine, it is alkaline in nature. Gram-negative bacteria are present here. This is large intestine. The large intestine or colon contains the largest number of bacterial community. It acts as the fermentation vessel, and the microflora consist primarily of aerobic gram-negative, gram-positive, non-spore forming, spore-forming bacteria. The facultative aerobes are also present here, and hello. Hello, am I audible?
Yes, absolutely. You are audible. Carry on.
This slide is about genetical tract. Mycobacterium smegmatis, a harmless commensal, is found in smegma both of male and female. Aerobic bacteria. Aerobic and anaerobic bacteria can be present Lactobacillus, G. vaginalis, alpha hemolytis streptococci, and C. trachomatis, and Ureaplasma urelyticum. The female urethra is either sterile or it contains few number of gram-positive cocci. Last we came to conclusion. A healthy human body is regularly exposed to the harsh environment which is full of microorganisms. A new project said that each person's microbiome is different and unique. Therefore, two healthy persons' microbial communities is different and they are still healthy. Acknowledgement. I would like to express gratitude to the organizing committee and Mr. Bishwajit Shah, Department of Microbiology, Vijay Krishna's College, for the support and my support. Thank you.
Hello. Thank you, Deepanita.
Yes, ma'am.
Thank you, Deepanita. Thank you.
Is there anyone? Chinmoy, are you join করেছে?
No one is left so far. Actually, now I will. দিদি আর কিছু বলবেন?
না খুব ভালো হয়েছে, অনুষ্ঠানটা খুব ভালো হয়েছে। এবার তোমরা এটাকে তোমাদের কি কাজ আছে।
So I will request Dr. Papiya Ghosh, Head of the Department of Zoology, Vijay Krishna Girls College.
স্যার অমিত স্যার ঢুকে গেছেন। অমিত স্যার মানে এন্ট্রি করতে পেরেছেন।
Okay. Then I will.
If Dr. Ghosh will give, I don't have any problem.
No, তুমি তাই জন্য আমাকে বলেছো। Okay, no. So then I will request Dr. Amit Mojumdar, please come and give the vote of thanks, please. Dr. Mojumdar.
Okay, actually, thank you, Mr. Chinmoy Vasu, our faculty of Department of Geology, to give me the chance to propose the vote of thanks. It is a great honor to have the opportunity to propose a vote of thanks to all who have helped us in making success in the fourth day of this second phase of webinar of the biodiversity organized by IQAC, Vijay Krishna Girls College, Howrah. I, on behalf of the organizing committee, extend a very hearty vote of thanks to the Honorable Chairperson, Dr. Moitrey Boshu Bishwas, Associate Professor, Geology, Uttarpara Raja Bihari Mohan College. She has contributed some valuable time from her busy schedule and made a great success in our today's webinar session. Thank you, ma'am. I also must mention our deep sense of appreciation to Mr. Chinmoy Vasu, Faculty Department of Geology, for his cooperation in this particular session. Nevertheless, I want to thank and congratulate all my dear students, paper presenters, and the participants for making the fourth day of the second phase of this webinar a grand success. I would also like to take the opportunity to congratulate the coordinators of the third day of the second phase of this webinar, Dr. Tanvir Nasim, Dr. Sushanta Shaduka, and all other members of this organizing committee. I also wish to thank Dr. Shweta Guha, IQAC Coordinator, Vijay Krishna Girls College, Howrah, for providing much needed encouragement and support. And the last but not the least, I would like to express my sincere gratitude to our Honorable Principal Madam Dr. Ruma Bhattacharya for her unending motivation, encouragement, and support throughout the journey. Thus, we are making up of today's webinar. We'll meet tomorrow for the 8th session at the same time at the same platform. Thank you all and good night.
Thank you. Thank you.
So I think we can formally close the session. I am requesting the organizer of this particular Google Meet platform, Mrs. Amrita Mukherjee, to formally end the session. Thank you. Thank you. Thank you, Amrita. Thank you, Chinna. Good night, madam. Good night, madam.
Okay, thank you. I'm leaving.
you.